Hello Ray.
Hi Ray, can you hear me?
Yep. I think I can hear you.
Yes,
Okay,
we
great.
can hear. Yes.
Okay.
Great.
Just want to make sure everyone can hear us before I start.
Yeah.
Hello, Jam. Hello, Ray. Hello, Ting Ting. How are you?
Hi, Dr. Loy. Hello.
Hello,
Doing great.
good evening.
Yeah. So I just want to make sure that because like I know Ting Ting is field application bioinformatics support from PacBio, and because like we have the student who come from background of wet lab scientists, so maybe they will have a lot of questions concerning about the wet lab. So I'm very happy that have Ray here. We'll have Ting Ting to answer some questions about the wet lab, like some kind of library prep preparation. Yeah,
Sure.
thank
I
you.
will try my best to answer. If not, I can collect all the questions and get back to them.
Yeah, thank you. Thank you. So it is the metagenomic course. It takes us like nearly uh, four 4 months for the first part of the short read, and now for the second part of the long read around 2 months. So every Sunday evening we have a lecture from 8 to 10 p.m. Yeah.
That's very nice of you, Dr. Loy, to give all these lectures. It's free, right? You don't get any.
Yes. Yes. So it's free from both sides. So for a student, I don't get any free, and for the um, platform provider or the distributor of the platform also, I don't ask. I just ask for help. Yeah, from you guys. Yeah. Thanks a lot for your time. Yeah.
Yeah, no problem. I think it's a good platform to create awareness of all these different applications and yeah, how it can help the research in Vietnam.
Yeah, and our Zoom we get from our friend who is a researcher in Darling Harbour in US. So and this link, the Zoom link only for maximum for 300 students. So that reason that we make another link in YouTube channel. Yeah. So
People can go back and look at it.
exactly. Yeah, we also have the YouTube link as well. Yeah. Okay, so now already 7 p.m. So I think that the first part of our seminar about the PacBio long read platform, especially we concern more about the long read in this time. Yeah, and our lecture is focused on the whole genome sequencing for individual sample, as well as we have like Metagenomics for full length 16S and shotgun Metagenomics as well. Yeah. So today we have the distributor DKSX from Vietnam, and also we
Recording
have
in progress.
PacBio. First we have Ray Clean here, and the second we have Ting Zhu. She is field application bioinformatics support from PacBio. Yeah. So now take it, Ting. I give everything to you now. Thank you again. Yeah.
Thank you, Dr. Loy. But you have to wait first. I will go first and then Ting will come next.
Okay, perfect.
Yeah, so
Yeah.
okay. All right, let me share my screen.
Can you please
One.
swap your display? Swap your display.
Yep. 1 second. Let me do it again.
Okay, perfect. We see the full screen.
Okay. All right. Okay. All right. Thank you so much, everyone, for spending your Sunday night with us. And thank you so much, Dr. Loy, for inviting us to come to this platform to introduce about our long read platform, as well as, how we can actually contribute to the microbiome research, as well as like microbial whole genome sequencing. Okay, so just to introduce myself, my name is Ray. So I'm the Territory Account Manager for Southeast Asia, for PacBio. So for my presentation, I will start by introducing about PacBio first, and then after that, I will go into the different applications that is being run on our PacBio platform for microbial research. And then after that, I will pass on to Ting to actually go through some of the tools to actually, do whole genome sequencing, shotgun metagenomes, as well as 16S sequencing. Okay, so I will start my presentation. Okay, so yeah, so just to introduce about PacBio first. So for PacBio, our mission is to actually enable the promise of using genomics data to actually better human health. And in PacBio itself, we are a company that actually have both the short and long read sequencing, so that we can actually offer the best technology for different types of applications. So for the long read sequencing side, we actually have two different sequencers right now, which is one is the Sequel 2E and the Revio, whereas for the short read sequencer, we have the own source system. So for the Revio is our newest instrument that is just launched, I think 1.5 years ago, where you can actually sequence up to 1,300 long read genome, human genome in a year. Whereas for ONSO, it's our state-of-the-art short read sequencer, which can actually, generate more than 90% of the reads that are above Q40. And then in our R&D pipelines, we are also coming out with a benchtop long read, as well as a high throughput long read, as well as short read. So with this multiple, portfolios that we have for our platform, we are able to address different applications. For example, rare disease, cancer genomics, and as well as the focus of today's topic, which is microbial and metagenomics. And we don't only, is able to offer results just on the DNA level. So we can also, do on the RNA level as well as the epigenetics side as well. And we partner with different partners to actually offer end-to-end solution to our customers. So for example, we partner with Twist, 10x Genomics, Agilent, or even like Zymo for some of the microbial applications on the upstream workflow. And then we partner with people like Hamilton, Ticken for the automation part to scale the workflow. And as well as the bioinformatics side for like Google Health, QIAGEN, and NVIDIA on the downstream bioinformatics part. Yep. So just to go into some of the basic of our long read sequencing. So today our topic will be mainly focused on long read, right? So our basis of our long read sequencing is called hi-fi sequencing. So what can hi-fi sequencing generate is reads that are both long, up to 25 kilobase pairs, and highly accurate, up to 99.9% accuracy. So how do we generate hi-fi reads is we will usually start from a high quality double-stranded DNA. And then what we will do is we will actually ligate the smart bell adapters on the both sides of the double-stranded DNA. And then we will anneal the primers with the DNA polymerase. And then after that, we will actually sequence this circularized DNA in repeated times. So we will sequence it multiple times to actually generate a lot of these different sub-reads. And then we will collapse all these sub-reads together to form a consensus sequence called a hi-fi reads. And with this hi-fi reads, it's, like what I mentioned, it's 99.9% accuracy, which is, the most accurate long read you have right now in the market. And it can actually generate reads that are up to 25 kilobase pairs. And yeah, just to reiterate my point again. So, our long reads, we can generate reads that are 10 of kilobases in length. It's 99.9% accuracy. And then because we are actually sequencing the individual DNA molecules itself, right? So there's actually no bias in terms of GC content from all the PCR processes. And because of this, we are also able to get the DNA methylation information without any special library prep. So you don't need to do any bisulfite sequencing to actually get the 5MC data. So, because we are actually directly sequencing on the DNA molecules itself. Yeah, and just to, reverse back to this platform itself again, just to show you, this is our latest sequencer, which is called the Revio. So for the Revio, we can actually generate up to 360 gig of hi-fi yield per run, which is 4 human genome. So we have 4 different stages, which each stage can actually generate up to 90 gigs of data, which is a 30x human whole genome. And each of the sequencing run take up to 24 hours. And like I mentioned, the read length that you generate will be around 15 to 20 kilobase pairs, and it will be 99.9% accuracy, which is Q30. And you will get the 5MC data together from the sequencing run itself. Yeah. So, just to, before I go into the microbial genomic side, just to, reiterate some of the trends, not only on microbiome, for, with our current hi-fi sequencing, we can actually, generate whole genome sequencing data that is without any gaps from all the, a lot of the structural variations, repeat expansions that were not able to sequence properly on the short read sequencing. But now on hi-fi sequencing, we can actually, sequence through those dark areas. And the data that we generate are also in collapsed haplotypes where you can actually, separate the paternal and also maternal allele. And like I mentioned, you will get your methylation information directly from the sequencing data. So you don't need to actually do bisulfite sequencing to get those epigenetics information. And on the RNA side as well, we do have our Kinnex workflow, which can actually offer you isoform level information, both on bulk RNA as well as single cell RNA. And of course, lastly, on the microbiome side, for our 16S sequencing, because we are doing full-length 16S sequencing, we are able to go down to species and strain levels compared to V3, V4 sequencing on the short-read sequencer. And on the shotgun metagenomic side as well, if we are doing shotgun metagenome assembly, we are able to generate more max or metagenome assembled genomes compared to short-read sequencing.
Hi, Ray. Can I have hi, Ray. Can I ask a question
Yes.
as previous slide?
Sure.
I have question about can I sequence the native on a full length on a from your kit?
Not yet. No. So for our RNA workflow is still based on cDNA. So we cannot sequence the direct RNA directly yet. But if let's say you do want to look at some of the RNA modification, right? We do have some papers that some researchers did do some pre-treatment during the library on the cDNA side to enable them to actually identify those RNA modifications. I can actually share some of the papers to you if you are interested.
Yeah, thank you.
Okay, so going into microbial genomics, right? So with our sequencing reads that are long as well as highly accurate, like you can actually run a broad range of applications on for microbial genomics on hi-fi sequencing itself. So mainly, there are four primary ways that you can actually use PacBio hi-fi sequencing. So the first one, of course, is microbial whole genome sequencing. So with hi-fi sequencing, you can actually get complete microbial genomes with ease and confidence. And for most microbes, right, on our sequencer itself, you can actually get a closed genome as well as the accessory plasmid all assembled with just one touch using the default settings on our assembly pipeline. So you don't need to actually use short read to actually polish the genome itself as well. And if let's say you're doing viral sequencing, we do have some targeted approach that you can actually use as well. And for 16S, so of course for the 16S, like what I mentioned, we are doing full-length 16S sequencing. So we can actually go down to species and strain level so that you can actually look at your true diversity of your samples itself. And for hi-fi shotgun metagenome profiling, so for our hi-fi sequencing, right, you can actually get, you have better recalls and precision on the on our hi-fi sequencing itself, and then you can also get richer functional information as well. And for finally, on the shotgun metagenome assembly, so with hi-fi sequencing, you can actually obtain many high-quality mags, and it's complete and closed genomes. And later on, I will show you more case studies as well. Our running on hi-fi sequencing is actually more cost-effective as well for shotgun metagenome assembly compared to other techniques. Okay. So I think there's a myth, right, that everyone, if I ask everyone on the floor, people will say, for PacBio, hi-fi sequencing, long read, it's actually, it must be very expensive, correct, compared to, running it on Illumina or even MGI. So I think this is the perspective that we have from the market itself. But I think this particular statement is now outdated because, with this year, right, we actually launched, a few different workflows. For example, like our Kinex 16S workflow, as well as our hi-fi plex prep kits, which actually, enabled new capabilities in the microbiome workflow itself. So right now, you can see that, for our Kinex 16S, if you compare it with short read sequencing, if you're running on high throughput sequencing, you can actually achieve even cheaper price compared to what you are running on the NextSeq itself. And then as well as on the microbial whole genome sequencing, as well as the shotgun metagenome side, if you are using our newest hi-fi plex prep kit, and on the Revio system itself, you should be able to get, even lower cost compared to what you're doing on a NextSeq. Okay. All right.
I just want to know, sorry, can you come back to the pricing?
Yeah, sure.
This one is only for the sequencing, not include library prep and DNA extraction.
So it includes, so for example, like the Kinex, so for example, 16S. So how we calculate this price is based on the Kinex 16S kit itself, as well as the sequencing costs. Because we are able to actually multiplex a lot of samples in one single smart cell. So that's why we are able to actually, bring down the price. So it includes the library prep as well as the sequencing, but not the extraction part.
Okay, thank you.
Okay, but just a disclaimer, so these prices are based on the US list price. So there might be some discrepancy in terms of the price in Vietnam itself, but this are references from in the US. Okay.
Yeah, thank you.
All right. So, just to, going back to the platform itself. So for our Revio system, as I mentioned earlier, the Revio has 4 different stages, right? So you can actually run, different types of applications on the Revio itself. So on the different smart cells. So for example, you have a run that is looking at, you have an experiment that is looking at 16S, as well as you want to look at their metagenomes as well. And then you do have some, collaborators that are looking in viral or microbial whole genome sequencing. You can actually run, multiple different experiments in one single run on different smart cells on the Revio itself. And of course, if let's say, you are doing other things like human as well, you can also, replace some of the different smart cells with other plant and animal or human application also. So with the Revio system itself, with the increased chip density as well as the lesser time required, so we are able to, increase the throughput if let's say you are running shotgun metagenome or shotgun metagenome profiling by, almost 10 times compared to the previous model that we have. So this is the reason, why the cost also goes down, because, we are able to actually run more samples on one smart cell. Okay.
I have a question, which means
Yes.
that you have I can run some application on single mast cell. I mean single chip. It means
Sorry,
that I can sequence 16S and metagenomics and microbiome whole genome sequencing just only one chip, one flow cell. Only
in this particular example, it's different application on different smart cells. But we do have customers that actually they try to run different application on one smart cell itself. So let's say you have some 16S and you have some metagenomes data as well. So we just need to make sure the library size are the same. Then you can actually run it on one single smart cell. But we usually recommend customers don't mix libraries together in one smart cell. Yeah.
Yeah, thank you.
Okay. So next we will go into the different applications to just further elaborate more on the 4 different applications that I described earlier. So the first one is of course on the microbial whole genome sequencing. So I think currently most of the whole genome sequencing for microbes are still being performed on the short-read sequencers, right, to actually produce a draft genome. So usually these draft genomes will actually have a few disadvantages. For example, there are poor resolutions of regions that are in terms of like structural variations or like mobile elements such as plasmids, and they might have genes that are fragmented and the genes cluster are not unassembled because of the the short read sequences itself, right? And then on the short read sequencing as well, there are no information of the RM motifs as well. So all these different disadvantages will actually give you consequences. For example, if you're not sequencing through all these different regions and elements, right, you cannot actually characterize, for example, traits such as antibiotic resistance or the transmission chains itself. And if let's say the genes are not sequenced through properly and it's fragmented, you might inhibit discovery. You might not be able to identify that gene itself, and you will overlook all these drivers of these key traits. And on the RM motifs as well, you will miss the contribution of this DNA modification, this impact of DNA modification on pathogenesis, virulence, transmission efficiency itself. Yeah, so by using hi-fi sequencing itself, because of the hi-fi long read itself, and it's highly accurate also, we are able to actually sequence through all this difficult to sequence regions, repetitive regions, as well as genes that are, because we are 15 kb, right? So we will usually sequence through the genes itself, and like what I mentioned, like the epigenetic side as well, we are able to actually look at the RM motifs also. And with PacBio hi-fi microbial whole genome sequencing, you actually do not need short read polishing. Yeah, so this is a very busy slide, but I just want to summarize to you to tell you that on PacBio itself, we do have an end-to-end workflow for microbial whole genome sequencing. So from the we have our nanobind extraction kit that can actually help you to extract high molecular weight DNA. And then we work together with our compatible partners for shearing, for example, things like plate-based high throughput shearing, Hamilton pipette shearing. And as I mentioned earlier, we have our hi-fi plex prep kit, that we can actually multiplex up to 384 samples. And depending on your throughput, this particular kit can be actually automated on the liquid automation platform as well. And then we can actually run, after we prepare the library, we can run the samples on, either the SQL 2E or our Revio platform. And then our Smart Link, which I think Ting will explain further more later on, that we have a pipeline to actually help to, perform microbial genome analysis. Okay. So by doing that, we are able to identify all these strain characteristics, the MR genes, as well as the mobile vectors mediating spread. Yeah. So just to show you all an example of comparison between short read as well as compared to using PacBio hi-fi sequencing to perform a whole genome sequencing. So this particular case studies was from one of our customers, and they are actually looking into some of the antibiotic resistance bacteria from patient samples. So they use our PacBio sequence long read sequencing to actually perform whole genome sequencing, and you can see that on PacBio long read we are able to actually sequence through a whole a closed genome, a circular closed genome, as well as the plasmid at the site. Whereas for the short read sequencing, you can see their contig assembly graph is very complicated because of the nature of the short read, right? So it has more challenges to actually combine all these different contigs together to form the genome. Whereas for a long read sequencing, we are sequencing through the entire genome itself. So that's why it's much more easy to have the contig assembly graph is much more easier to actually look at. So with this particular contig this particular genome that looks like this, it will also impact your downstream analysis as well if you are looking into plotting things like phylogenetic trees. And another example is another paper from one of our customers doing sequencing through the MTB or Mycobacterium tuberculosis complex genomes. And they actually saw that with just 16x, it was sufficient to actually obtain a closed genome. And the accuracy of hi-fi reads allow them to actually forego many of the complexity of the methods from previous approaches. So noisy long, if let's say you're using another long read, which is a bit noisy. you were required to, sorry, excuse me, I'm back. Okay. So for things like, because of our accuracy of our hi-fi reads, right, we can actually, forego many of the complex methods that were used for previous approaches. For example, you have to use hybrid approach to combine long read as well as short read together to do assembly. But because of our hi-fi reads are so or are already accurate, right? So we don't actually need to use the hybrid method. And in this particular paper itself, you can see that they are able to actually identify duplications that happened in the MTB genome itself, and the duplication is like 7.2 base pairs long. So you actually have to sequence double of this, which is around like 15 kb, to be able to identify these duplications. So I don't think short read will be able to identify this particular duplication itself. Yeah, and also PacBio sequencing is also being used as part of the surveillance pipeline in the US as well. So in one of the hospitals in the Boston area, they actually use our hi-fi sequencing to be like a second line. So they actually if there's any novel strain of this particular carbapenem resistance enterobacteria, they will actually use our PacBio sequencing to sequence through the novel strain to identify, the strain characteristics, AMR genes, as well as, any of the mobile vectors mediating spread. Okay, so that is on whole genome sequencing. And then I will go into the metagenomics. So metagenomics means, looking into microbial communities. So I think 2022 and 2023 was a breakout year for hi-fi metagenomics. There has been, multiple publications that, uses our long-read sequencing to look at both 16S as well as shotgun metagenomics using hi-fi sequencing. So the number of papers that you can see, after like 2022 and 2023 and 2024, the number of hi-fi metagenomics publications have been increasing by a lot. I think one reason is, of course, researchers are actually see the value of our long-read sequencing for metagenomics, as well as, of course, the declining cost of our sequencing output itself. So that's why more and more people are actually jumping into using hi-fi sequencing for metagenomics work. So the first metagenomic technique that I want to talk about is, of course, 16S sequencing. So we know that for 16S, all bacteria actually have a 16S gene, right? And usually we try to identify the variation in this conserved genes so that we can actually enable we can actually identify the species or genus of this particular bacteria itself. So targeted sequencing of this 16S is actually a much cost-effective way to actually assess the composition of the metagenomics communities. So I think usually for short-read sequencing, a lot of people are actually still looking at just V3 and V4 region, sequencing this particular region. But for hi-fi sequencing, we actually advocate to actually do a full-length 16S sequencing. So because by doing full-length 16S sequencing, you are able to actually detect more nucleotide differences, for better taxonomy resolution. And we can actually distinguish, down to species and strain level compared to, just looking at V3 and V4. Yeah. So, just to show you a publication from George Weinstock, one of the leader of the Human Microbiome Project, right? They actually showed that, looking at different regions of the 16S gene itself is biased towards certain bacteria. And for example, for V4 itself, if you look at this particular image here, right? If so the redder you go, which means the more unclassified percentage, unclassified microbes that you have, unclassified bacteria that you have in your sample itself. So you can see that for V4, there are many, it's so red, which means, many of the bacteria are actually unclassified. Whereas if you are doing, a full length, for example, V1 to V9, you can see that most of the bacteria is you can actually classify them into the clade itself. Yeah, and you can see that there is also another paper that actually shows that they will actually comparing between PacBio full-length 16S sequencing compared to V4 as well as synthetic long-read full-length 16S. Means you use short-read, but you combine them, you sequence through, and you combine them to actually form a synthetic 16S. So it shows that there's 99.7% of the sequences can be assigned to species level when we are using PacBio hi-fi sequencing compared to loop-seq, which is the synthetic long-read full-length 16S, and also V4 itself. And there's also another publications that have shown that there's a high correlation between full-length 16S compared to shotgun metagenomics. So in this particular paper itself, they showed that they found 140 species classified in both data sets, and 88% of them assigned to 16S and 74% of them assigned to the shotgun reads. So and even at genus level, the correlation actually increased to 90%. So by doing full-length 16S sequencing, you have data that is similar to what you can get from doing shotgun metagenomic profiling already. And some of the interesting applications, of course, we know that people are doing gut microbiome as well as human gut microbiome, and we do have customers that are using our full-length 16S to do animal biome as well for dogs. So they do have kits that they've developed for dogs as well as for cats to actually look at their gut microbiome health itself. So I think this company is in Australia, and we do have this paper. So if you are interested, we can actually share to you. So for 16S itself, right, we do have two different techniques that you can use. One is on the standard monomer protocol, whereas the other one is the Kinex protocol that I mentioned earlier, which actually make our cost of 16S to go as low as the short read sequencing. So for standard monomer means we are actually sequencing just one 16S gene in one single SMRT well in the smart cell itself. So this is suitable if you are not running a lot of samples. But if let's say you are running more than 192 samples on the Sequel IIe or 384 samples on the Revio, we will recommend you to use the Kinex workflow. So what the Kinex workflow does is it will actually combine. So we know the 16S the full length of the 16S sequence is around 1.6 kb, right? But as I mentioned, our hi-fi sequencing generates reads that are up to 15 to 25 kb. So if, for every well, we are just sequencing 1.6 kb, we are wasting a lot of the potential that you can actually sequence, right? So what we do in this Kinex workflow is we actually concatenate, or we combine multiple 16S fragments together to form one single, long read itself. And then this one single long read, we will actually, we will prepare this library, and then we will actually subject it to sequencing. And then after that, we will use bioinformatics pipeline to, demultiplex them and, to generate the each segmented reads itself. So by having this Kinex 16S kit, we are able to actually, increase the throughput by 12-fold. And by doing that as well, you are able to actually, save a lot of cost in terms of you can multiplex a lot of samples on one smart cell itself. So that's why we are able to actually generate, the low cost, which is, as that is, comparable with what you are running on short read right now. Okay, then I will speed up a bit so that So next is on the shotgun metagenome profiling. So we know that, for shotgun metagenome profiling, our goal is to actually identify the species that are present in a sample. For example, a soil sample or a fecal sample. And also we want to know the relative abundances as well as look into, you know, some of the functional information or gene information in that particular sample itself. So hi-fi reads, there have been, you know, many of the publications which I have shown you in the one of the slides earlier as well, where, you know, a lot of researchers are actually using our hi-fi for hi-fi metagenome profiling, because we can actually assign taxonomy with high precision and recall. And, you know, each hi-fi reads, because we are, you know, so long, right? You can actually have, you know, multiple genes intact per read, which can actually provide you richer information about the genes that occur in single microbe and maybe co-express as well. So 80 or to 90% of the hi-fi reads will have at least one functional annotation, where I can show you in one of these case study by Soltea Therapeutics. So they were actually doing a comparison between they were running these fecal microbiome samples on both short read and long read sequencing. And they found that, you know, only 34% of the short reads can be annotated. I mean, it's logic, right? Because, you know, short read is only 100 to 150 base pairs, right? So genes are usually, you know, longer compared to 100 to 150 base pairs. So that's why, you know, only 34% of them can be annotated. Whereas for hi-fi reads, right, you know, up to 70% of our hi-fi reads can be annotated. And oops. Yeah, and, you know, each hi-fi reads actually has an average of 4 annotations, where, you know, short reads have at most 1. So you need actually, you know, 10 times fewer hi-fi reads to generate the same number of functional insight compared to what you're doing on the short read sequencing. Okay, then next is on the last applications that I would like to just further elaborate is on shotgun metagenome assembly. So for the short metagenome assembly, so just now metagenome profiling is we want to actually profile to see, you know, in a microbiome community, how many, you know, what species are in that community itself, and also, you know, what are the relative abundance and also the gene function or the functional information, right? Whereas for metagenome assembly, we will usually sequence deeper, so that we want to actually, you know, assemble a representative genome from this microbiome sample. So instead of, you know, doing, you know, pure culture and doing microbial whole genome sequencing for a single culture, for metagenome assembly, we are actually trying to assemble the genome in a microbiome sample itself. Okay, but there are certain challenges, of course, right? Because, you know, there are multiple species that are present, and there's uneven and unknown coverages. And of course, there are things like difficult regions as well in these microbe genomes. So if you compare between the short read metagenomics and if you compare to what we are doing on hi-fi itself, right? So as I mentioned in earlier as well in the microbial whole genome sequencing application. So because our reads are are long and accurate itself, right? So once we sequence through the you know, microbiome sample itself, we are able to generate reads that are very long, and some of them, you know, will actually, you know, already form like a closed genome itself. So by doing that, we can actually, you know, form complete genomes, and genomes that have lesser gaps compared to what you have on the short read side. So for the short read side, you rarely will be able to get a whole genome of a bacteria based on, you know, 100 to 150 base pairs, right? You will still need to use bioinformatics tools to actually assemble them to get the full genome itself. But a lot of time, you know, because their reads are so short and then there are a lot of complex regions, you'll not be able to get a full genome, and there will be a lot of gaps in the genome itself. And of course, compared to another another long read technology itself, right? So we know that, you know, metagenomes contain many closely related strains, and some of the closely related strains only have a difference of only about like 3%. You know, 3% is a common cutoff for defining a distinct species. So if the difference between the closely related strands is, you know, is less than the error rate itself, you know, maybe less than 3%, the error correction can actually erase these real differences. And then you might miss those strands itself. You might call them as the same species and same strand because of the error correction that has, corrected the error away. Whereas, for hi-fi reads, because we are 99.9% accurate already, you do not need any error correction. And we can actually be able to actually assemble the genomes properly and to be able to resolve strain level information and have a complete genome itself. Yeah, so just to show you a paper that, compare between the Nanopore technology as well as the PacBio Hi-Fi technology with and without short read polishing. So if you look at the darker green or the darker colors, right? So darker green, darker yellow, darker purple. So these are all the sequences that are just based on the pure long read sequencing itself. Whereas the lighter ones are the after the researcher polish it with the Illumina data. So you can see that, for PacBio data itself, right, with or without polishing, our ideal score is so much more higher compared to what ONT can actually offer. And some of them, you can see that, the score actually dropped down with the polishing itself. So this is the reason why we are saying, we actually don't need short read polishing for most of our whole genome sequencing as well as the shotgun metagenome assembly pipeline. Yeah, and lastly, just to show some of the recent preprint as well, comparing the different technologies. And, for our PacBio data, we actually yield up to 70 times more complete metagenome assembled genome, or max, compared to Illumina, or 2.5 times more than ONT itself. And our PacBio outperforms ONT in all the key metrics, for example, read quality, mean median read length, and 50 read length. And our max usually have higher completeness and lower contamination. And in terms of cost as well, because of all the different, prep workflow improvement and platform improvement that I mentioned earlier on as well. So the researchers actually, calculated that the cost of running PacBio is actually the most cost effective. And another comparison to show you, comparing hi-fi reads versus short reads in one of the paper looking at soil microbiome. So you can see that, in this particular paper, they generated around 629 gigs of hi-fi data, whereas they generated around 3,400 gigs of Illumina data. And you can see that the number of max that they actually are able to actually achieve in the particular sequencing run. So you can see that for PacBio data, they are able to generate up to a 2,041 max, which 587 is a single contig high quality max, and 315 is high quality max, and 1,000 of them are medium quality. So this is, you can see that is the differences between the data that you can get from hi-fi reads and short reads are so significant. So for Illumina, they are only able to get around 307 max in this particular study itself. So if we calculate, the consumable costs and we divide by the number of max itself, you can see that, for each of the max that we actually achieve, we can actually, it's only cost $5 on PacBio. And for high quality max, it costs around $11 on PacBio, which is, such a big difference compared to what it is to run on the short read platform. Okay, so this is my final slide, just to show you all, we do have, all the different workflows and different protocol for, for full-length 16S, key-neck 16S, as well as if, let's say, you're looking at fungal or eukaryote amplicon profiling, we do have application node to help you all to do that. And then for shotgun metagenomics as well, we do have our PacBio whole genome sequencing library prep workflow that you can actually follow to actually, perform these experiments. Okay, so with that, I think that's the last of my slide, and I will pass it on to Ting to go through some of the tools that we have for microbial and microbiome research from PacBio.
Thank you, Ray. Maybe you please share the screen again. Because we really want to discuss about the web lab, and then we
Sure.
move on to the bioinformatics later.
Okay.
So I will starting with a question, and then you guys will come up later, please.
Can you see my screen? Yes.
Yes, I can see. So what is the difference between the protocol from the PacBio full-length 16S and the kinase full-length 16S? They two different, totally different two protocols are the same.
Yeah, so for the difference between the PacBio full-length 16S and Kinase full-length 16S is there's an additional Kinase protocol on the full-length 16S part, where we actually concatenate the 16S genes. We concatenate like 12 different 16S genes in one single read, so that we can actually increase the throughput, which I will show you here. Let's see this one. So this is the 16S Kinase 16S protocol. So for the normal standard monomer protocol, right, what we do is we will just amplify the 16S region, and then we will sequence directly. But as I mentioned, right, the 16S is only 1.6 kb long, right? So if we put it into PacBio sequencing, we are actually wasting a lot of the the things that we can sequence. So what this Kinase do is we actually combine multiple 16S fragments into one single long fragment. So we combine, I think, 12 different 16S genes, 16S fragments together into form one long read, and then we will sequence it through. So by doing that, we are able to actually increase our throughput and lower our costs.
Okay. So it means that we save a lot of money when we use the Kinase to using the standard protocol.
No. So because you are spending a little bit of money on the Kinase kit, but it combine the reads together, and then you are able to multiplex more samples, and then you will be able to save on the smart cell itself.
Okay. Okay. But we have to pay for the kit of Kinase, is it? For lab preparation.
Correct.
Yeah. So it means that if you have a trade-off between them, yeah. Okay. So and then my second question concerning about the equipment required for doing long read, because like in the extraction or in the QC for the long fragment size, ready for the long read sequencing, do we have to, does it require any equipment specific to repair library for the long read PacBio?
Yep. So we will recommend for QC instruments, for example, like the femtopulse, as well as the qubit, as well as the bioanalyzer. So for our PacBio sequencing, we always say that if you don't do proper QC, right, if you don't do a QC check to make sure that your library is built properly, or your extracted DNA is high molecular weight itself, right? So if we always say rubbish in, rubbish out. So if let's say your DNA that are extracted is not high molecular weight, and then you don't do a QC, right, and then you go through library prep, and then library prep will cost, library prep reagents will cost, right, and then smart cells will cost as well. So we want to make sure you actually QC your samples properly, so that before you move on the next stage, so that you don't waste the reagents itself. So you will need femtopulse to check on high fragmented DNA, qubit to check on the concentration of the DNA itself, and then nanodrop to look into the A260 A280 ratio to look at the contamination itself.
Okay. So do we have any special treatment to keep the fragment size in longer to do the shotgun metagenomics? Do we need any special treatment for the DNA to make it longer, the fragment size?
Yeah, I understand what you are saying. Just that I'm thinking how to answer this, because I think the the issue with shotgun metagenomics is on the sample type itself, right? Because we know that, the samples can be coming from soil or like coming from blood or something else. So of course in PacBio itself, we do have our nanobind extraction kit, which is suitable to actually extract high molecular weight DNA from things like blood as well as insect and from human samples. And yeah, but I think for soil samples, things like that, our nanobind kit does not have the protocol yet, but I think there are kits from like Zymo or that is suitable for extracting those difficult to extract samples.
Okay. So have one question from Thảo Cao. She asked that E16S full length PacBio sequencing sufficient for the strain level classification, because she tries one data from NCBI. The data come from the 16S high five PacBio. However, the data only achieve the species, but
Species level.
species level only. Yeah.
Yep.
But however, the species level also very good, but she concerning the strain, the subspecies. Yeah.
Yeah, I think some of them will achieve species level and some of them will achieve strain level. So not all can achieve strain level depending on the specific, whether the 16S whether the difference in nucleotide on the 16 for the particular bacteria itself, whether is it on identifiable from the 16S itself or it's maybe on the other region. So maybe 16S is not sufficient enough to look at strain level.
Okay.
Yeah, so not all can go under strain level. So we just say, compared to short read, short read usually just give you on a genus level, but for long read, we can achieve, species or strain level. Yeah.
Yeah. And then have one more question about Huy Hà. He asking, does the time taken for library preparation step is different between different kind of sample, like soil, like water, like blood, like wastewater?
I think the difference will come from the extraction part. The extraction protocol will be probably different, but once you get high molecular weight DNA, the subsequent step will be the same.
And then I'm so curious about the timing between the standard for full length and then the kinect for full length 16S. The time preparation is different or not?
Yes, definitely there's some difference. So the process, of doing kinects, right? There's I think around a 2 days or 2 to 3 days workflow to actually do the kinects workflow. Yeah, but because for 16S itself, it's very easy, right? You just amplify this particular 16S region. You have the 16S primers to actually amplify this, and then you can actually do the library prep to stick the smart belt adapters onto it and run PacBio sequencing. But for kinects, because there's a concatenation process that is required, so there's an additional 2 days workflow that is needed.
Okay, thank you. And then
I have question for this
please.
slide. How many symbol I can navigate the 16S DNA?
So in one kinects protocol, we will ligate 12 16S fragments onto one long read.
And I can ligation the 16S in meta symbol. I just ligation 16S on I just use kinect 16S on in some kind of symbol like pill culture. I mean that the bacterial is isolated from the mixture.
I think the if you how to decide whether you want to do kinects or not it depends on the throughput. So it doesn't depend on the sample type that you have, but it depends on the throughput that you have. So if you have a Revio, if you're running less than 384 samples per smart cell, we will recommend you to just do the standard monomer full length 16S. But if let's say you have more samples, like more than 384 samples on the Revio, then we will advise you to use Kinex. Then it will be more cost effective on Kinex. If it's lesser than 384, right, running Kinex might be a bit wasteful. Yeah, you might have too many reads for one sample.
Thank you.
Can I please have a question?
Yes.
Do you treat different types of bacteria different way? For example, gram-negative and gram-positive differently when you do the DNA extraction? That's the first thing. And the second question is that how do you know in the mix of culture and a mix of bacteria that going through metagenomics that you already sequence every single one? Like, for example, you have stool sample or you have any kinds of mixed bacteria, and you know that you detect every single one in there. Thank you.
So I think for for like whether you are saying whether how confident we are that we sequence everything that are already in the fecal sample itself, right?
Yeah.
So I think this depends on the number of reads that you actually put in. So I think the more reads that you actually put in, for example, for 16S sequencing, you are able to look into more in depth of the diversity itself. But if let's say 16S is not enough to look into all the different species of bacteria in the sample itself, then you might want to look into doing shotgun metagenome itself. Shotgun metagenome will of course give you more information compared to 16S. So it's not only bacteria, but all the other things as well. Yep.
It's okay, Han. On
Yeah,
board?
thank you.
Okay, perfect.
Actually, I was asking about
Extraction.
do we need to treat the different types of bacteria differently when we do the DNA extraction? For example, gram negative, gram positive, because I know that gram positive have a the world cell is more difficult to extract. So do you have different kits for different kind of sample like that?
We don't. I mean, PacBio don't, but I think our compatible partners should have like this kind of like extraction kit that are suitable for metagenomics lah. I think like from Zymo, they should have kits that are suitable for metagenomics extraction. Or I think Hygen should have too. Yeah.
Thank you.
So, and then I have one question from Hoàng. So, he asked, what are the advantages of using PacBio for meta assembly, and what are the disadvantages that we should consider when doing the metagenome assembly?
I thought my presentation is all talking about that. No, but it's yeah. So if you're using PacBio Hi-Fi to actually do metagenome assembly, right? Of course, as I mentioned, with PacBio Hi-Fi, because our reads are long and accurate, right? You are able to get all this high quality max. All this closed genome. So when you do the metagenome assembled genomes, it's all you can see it's like, you have closed genome and lesser gaps compared to when you are running short read sequencing itself. So for short read sequencing, there are regions that are difficult to sequence. There are, and they will have a lot of gaps on their max itself. And I think I've shown, the publications, right? If you are using PacBio Hi-Fi reads to actually do metagenome assembly, we usually see, our technology actually generate more max compared to, even ONT or Illumina. And the most significant one was this one, like, by using hi-fi sequencing, you can see that, with our, little amount of number of data, we are able to actually able to actually get so many max compared to, Illumina with like, 3.4 terabytes of data, and they are only getting like around like 600 max. So that's the benefit of using PacBio.
And then what should we consider when we do it? There any disadvantage or before we should notice before proceed with the experiment?
I think the most challenging part, like I think a lot of questions that have been coming through for metagenome is of course the extraction part. Like, a lot of these soil samples, or they contain a lot of inhibitors, and you have to actually, pre-treat them properly to actually, extract high molecular weight DNA. So I think that is the challenge that we should be having.
Okay, thank you. And then you show me some commercialized industry like the vet for evaluate the microbiome for the cat and the dog. There any industrial application of PacBio for microbiome?
I think
Like
a lot
in
of
food
the
industry.
yes.
Mm-hmm.
Yes, so I think we do have some customers, for example, in Taiwan, that they actually do 16S for they are using more for human microbiome studies using 16S. So they actually do like a health screening for patients that come in for health screening. They will offer them our PacBio 16S sequencing to actually look into their gut microbiome, so that they can actually provide some kind of recommendation in terms of probiotics or diet change. And we do have labs in Thailand that actually they actually offer 16S for food security as well. So like things that they want to actually look into some of their manufactured food, right? So they will sometimes run 16S on our PacBio sequencer also.
So there is any CIBD for the application in Taiwan, as you said?
No. So currently all our solutions are research use only. Yeah,
Okay.
there's no IVD for it.
Okay.
Usually customers will do certain types of validation, and then they can actually launch it as a test, as a screening test usually. Yes.
Okay, thank you. So any further question, please? So again, the distributor of the PacBio in Vietnam is from DKSH. So DKSH là nhà phân phối PacBio Việt Nam. Nếu mọi người có liên hệ gì thì có Trâm ở đây ha. Trâm ơi, em đánh vào cái địa chỉ contact ha Trâm ha. Rồi, so now we move on to the next part about the bioinformatics analysis for the PacBio long read sequencing. Ting, please check it.
Yeah. Okay, let me share my screen. Okay. Yeah, I guess you can all see my screen, right?
Yes, I can see your screen now. Thank you.
Okay. So good evening, everyone, and welcome again to for joining today's webinar. And so I would like first to thank Ray for introducing the advances in sequencing with PacBio hi-fi reads, both on the data quality and the cost. And in the following 30 minutes or so, I will tell you about how you can achieve those high quality results using the bioinformatics resources we offer here from PacBio. So here I'm showing you again the 4 primary applications using hi-fi technology in the microbiome space. So on the left, you can see microbial whole genome sequencing. So you do a culture and you perform isolate sequencing, and then full-length 16S sequencing, which allows you to get to species and strain level identification. And then the last two are both related to metagenomics, which is the shotgun approach. And in the following time, I will talk in detail on specific data analysis approaches for each of these 4 applications. So let's start with microbial whole genome sequencing. And with regard to the microbial genome assembly, we have this workflow, which is called microbial genome analysis workflow. This workflow is specifically developed for microbial genomes assembly. And with this workflow, you will obtain not only the chromosome assembly, but also the plasmid assembly simultaneously. And the output sequence has been formatted for NCBI submission, so you don't do any modifications on your sequence. You can just submit the output results directly to NCBI. And in addition, you can also specify the base modification detection if you would like to have that information. For example, you can specify to obtain 6MA or 4MC information using this workflow. And this microbial genome analysis workflow is available in our software SmartLink. So SmartLink is our official software, and it is implemented with a graphical user interface. So this will make things much easier if you are not familiar with the command lines, especially you're a wet lab researcher and wants to analyze the data by yourself. And here I'm showing you how this analysis workflow looks like in SmartLink. So it is just a push button workflow, and all you need to do is to indicate the data set you want to analyze, and then to determine whether you want to also detect this base modification or the motif-based motifs. And finally, you just hit this start button, and the analysis will perform automatically. So to show you the performance of this workflow, we prepared 96 libraries from 24 distinct bacteria relevant to food safety and infectious diseases. Those different bacteria has a wide range of GC content, genome size, and complexity, and each with 4 replicates. And we then sequenced and assembled them using this microbial genome analysis workflow. So here in the top figure, you can see that we have 92 out of 96 microbial genome assemblies with the expected chromosome numbers. And you can also see in the bottom figure that most of the bacterial genomes have the expected number of plasmids. And when we look at the assembly accuracy of these microbial genome assemblies, we found that these assemblies are all around Q50 or so. So that's one of the benefits you can get from using highly accurate hi-fi reads. And here I'm showing you a case study that we performed in collaboration with a customer. What they wanted to do is to evaluate the different sequencing technologies and how it works for their antimicrobial resistance, or AMR, detection pipeline. So here's the result on the contiguity of the assembly using ONT and PacBio data. So ONT data was shown in blue, and for PacBio data, they used 2 different assemblers. The first one is our microbial genome assembly workflow in SmartLink. So here we use MA for short, and the other assembler is Hi-Fi ASM. So the results from these 2 workflows are shown in different shades of magenta. So here black is the expected number of contacts. So you can see that compared to the expected contacts, PacBio outperforms ONT for most of these bacteria. And when looking at the accuracy, sensitivity, and the precision of that study, you will see that PacBio in magenta here outperforms both Illumina and ONT across the board, especially for accuracy and sensitivity. And then the customer fed their AI algorithms with the whole genome assemblies created with each technology to predict a bacterium's resistance or susceptibility to a specific antimicrobial compound. So as you can see in this table, PacBio data outperforms in agreement and accuracy while showing the least errors. And next, we will move to the full-length 16S sequencing. And we know that all bacteria have a 16S gene, and variation in this conserved gene enables clade identification. So this is a study from our collaborator that they wanted to see the differences between individual variable regions of 16S and how they're able to classify bacteria to the species level. And then they compared these results to those from the full-length 16S that include the whole set of V1 to V9. So their results showed that since the human gut can harbor a broad diversity of bacterial clades, so only full-length 16S species can provide unbiased resolution of all the species that may be present in the microbiome. And furthermore, they were able to identify SMP locations through facing full-length 16S gene sequences generated for each individual isolate. So here you can see that x-axis denotes position along the 16S gene, and y-axis denotes individual isolates clustered based on their inferred phylogeny. And the dark blue region, so these dark blue regions indicates the location of a polymorphism. So as you know, the short-read 16S sequencing looks at only the V3 to V4 region. So they found that 91% of cultured isolates are not distinguishable because of intragenomic SNPs. But on the contrary, full-length 16S with hi-fi sequencing can provide a wider range of polymorphisms for species and strain level identification. So to help you achieve the species and strain level identification, we developed an ASV-based workflow to analyze full-length 16S hi-fi sequencing data. So this is the overview of the PB16S NF pipeline, and you may find the detail in our GitHub page here. So you can just click this link to get to our GitHub page. And we have detailed documentation to tell you how this workflow, how to install this workflow. And we have also demo data set for you to test it, to play it around. And here I would like to mention, so after taxonomy classification, this pipeline will generate an HTML report and a visualization to summarize the key results and statistics. And most importantly, the output from the pipeline is fully compatible with KIME2. So we run this workflow across different types of samples and found high percentage of reads assigned at the species level. Although here you can see in magenta that the relatively low number of reads assigned at species level for some complex environmental samples. And this also highlights the need for sample type specific databases, which may improve taxonomy resolution. So we rerun the taxonomy classification against the MIDAS wastewater database. And then these two numbers increased to 85% and 91% respectively using the MIDAS wastewater database. So the next application is read-based profiling and classification. And as Ray has just mentioned previously, the goal is to identify the species present in the sample and estimate their relative abundances. But there are several challenges associated with profiling and classification. And the most important one is that a lot of the methods for long reads are still being developed. And so the best practices and how these methods performed was unclear. And there's also a difficult balance between precision and recall. So that is how we can minimize false positives while also being able to detect species at very low abundances. So to address these questions, we performed a comprehensive benchmarking study of profiling and classification methods, especially for long-read shotgun metagenomic dataset. And this study has been published in the paper shown on the right in 2022. So here I'm showing you the 11 methods we tested, and we were able to identify 3 top-performing methods, including sour mash, diamond and Megan LR, as well as bug-seq. So these are some, there are still some differences between these methods. For example, sour mash is chimer-based, whereas diamond and Megan LR is based on protein alignments, and bug-seq is based on nucleotide alignments. And one thing I want to point out about the diamond and Megan LR workflow is that in addition to generate a taxonomic profile, it also simultaneously creates a functional profile. That's currently the only method that's capable of doing that simultaneously. And we have built workflows for both sour mash and diamond and Megan, and you can also check them out in our GitHub repo. So now let's jump into some of the results of these 3 methods. So you can see the numbers of species detected at each of these different abundance categories. So in the top row, we have ultra low abundance, and towards the bottom, we have higher abundance. And what I want you to focus on first are these methods in gray, so which are the optimized filtered methods tuned for high precision. And we can see that we detect 150 to 55 to 223 species based on these filtering criteria. And in white, so these 2 columns, I'm showing some unfiltered results from these different methods. And what you can see is those numbers jump up quite a bit, up to greater than 7,000 species with no filtering, and which you will notice most of these assignments are happening in the ultra low categories. And based on our study, we've quickly identified most of that is noise. So this demonstrates that the importance of using this threshold for internal filters in these programs to obtain high quality confident results. And also here, I'm showing you some of the results from the artificially downsampled datasets using the diamond and the Megan workflow. So we have 4 smart cells of data down all the way to a 48 plex. So this represents a 90 gigabase of data down all the way to 0.5 gigabase of data. And from comparing the results, we not only see the same number of species, but also really consistent relevant abundances across total data. So this really opens the door for using shallow shotgun sequencing to generate highly accurate taxonomy profiles. And as I mentioned previously, the diamond and the Megan workflow can simultaneously output functional annotations in addition to these taxonomy profiles. And you can see that approximately 92% of the hi-fi reads received at least 1 functional annotation. And in total, over 67 million functional annotations were assigned across all the 4 databases. So this again shows the capability to build a highly accurate taxonomy as well as functional profiles using hi-fi reads. And here's another example showcasing that hi-fi data is rich in functional information. And in this table, you can see 5 human fecal samples. The mean hi-fi rate length is about 9 kilobases. So this means you can detect 8 to 9 genes in one hi-fi read, even without assembly. So in a scenario of genotyping your metagenomic samples, you can just go directly with the gene annotation, and there's no need to assemble hi-fi reads. And one exciting thing is that only 4 long-read methods available during our benchmarking study mentioned previously. However, over the last maybe 6 to 7 months, 6 new methods focusing on long-read taxonomy profiling and classification have appeared. So this really suggests that the research community is recognizing the power of long reads. So now let's move on to the last main topic of today, which is metagenome assembly. And the goal here is to sample a representative genome for each microorganism present in the microbiome sample. And we refer to these genomes as MAGs for metagenome assembled genomes. So there are quite a few challenges here. And first of all, there are multiple species present in the microbiome sample. And second, these species are present in uneven and unknown coverages. And finally, there are repeat regions and intergenomic overlaps that occur across species, as well as between strains of the same species. But the good news is, hi-fi reads can really help with this problem. So in the beginning of 2022, we published a new assembly algorithm called hi-fi ASM Meta. So this is specifically tuned for the hi-fi reads. And in the publication, we demonstrated that hi-fi ASM Meta produces more total MAGs and, more importantly, more circular MAGs than any of the other methods that are currently available. And in January 2024, another powerful tool called MetaMDBG is published in Nature Biotechnology. And we have done some work to compare these two assemblers with others. And our results showed that these two assemblers are currently the best practices for assembling MAGs. So just to give an example how hi-fi ASM Meta looks like, and here's an assembly graph from a human gut microbiome sample. And look closely, you will notice there are actually 75 circular contacts here. And these all range from about 1.2 to 6 megabases in size. So these are complete microbial genomes that come straight from the assembler. But you will also see that there are plenty of linear contacts here. And this is the reason why we would need post-processing pipeline to identify and extract these additional high-quality MAGs from this complex set. As such, we developed a hi-fi MAG pipeline, and this pipeline takes assembled contacts and outputs the high-quality MAG sequences, metadata, and visualizations. Now, the version of this pipeline is 2.0, which implements completeness-aware binning strategy. As a result, version 2 shows not only an increase in total MAGs, but also an increase in single contact MAGs comparing to the previous version. And so in this example, we run hi-fi ASM Meta and the hi-fi MAG pipeline on the same human gut microbiome sample. So we were able to recover 189 high-quality MAGs and classify them into 154 species. One of the interesting things is that 14 of these MAGs cannot be classified to the species level, and these are likely to represent a novel diversity in the human gut microbiota. In addition, there are 19 species that are represented by 2 to 3 MAGs, implying there's a strain level variation among them. And so here shown on the bottom, we obtained 2 representative MAGs from the same species and align them to the genome taxonomy database reference. So you can see in these dot plots figures here, there is still some structural variations across the genome. And that's even true if we compare the 2 MAGs to each other. So remember, these 2 MAGs are from the same species, and we can still observe those structural variations. And in the following time, I'm going to show you 2 pilot projects that we collaborated with 2 different groups. So one is the Banfield Lab at UC Berkeley, and the other is from DSMZ in Germany. As you may have been aware, that soil is so far the most difficult environment sample because it has the most diversity of microbial species, and many of them are in very low abundances. And therefore, it is very difficult to assemble all of the organisms that occur within those communities. So we performed a deep sequencing on 3 different types of soil samples, including wetland, woodland, and grassland, using our Ravio system. And we generated 3 to 4 smart cells on Ravio per sample. We assembled them using our 2 current best assemblers, Hi-Fi AS Meta and Meta MDBG. And then we conducted binning and QC using like the post-processing using our Hi-Fi Mac pipeline that's available on our GitHub. And finally, we also performed downsampling experiments to understand various data levels and how many Macs you can expect from those data. So here in this table, you can see we have about 200 gigs of data for the Banfield dataset. We have 280 gigs for DSMZ1 and 150 gigs for DSMZ2. And here's an example sampling graph from the wetland soil of the Banfield example. And you can see here a lot of circles that represent a complete chromosomes. And we actually have about 360 approximately of these circular contexts within this one sample. And these are all greater than 1 megabase in size. And we have one that's even super large here, which is a 13 megabase genome. So looking at the actual number of Macs recovered, we're getting hundreds of thousands of total Macs from all of these different samples. And many of them are single contexts, and as much as 50% of the total Macs are actually high quality Macs. So in this study, high quality means greater than 90% completeness and less than 5% contamination. And medium quality is 50% complete and less than 10% contamination. And more importantly, the Macs consist of very few contexts. So the medium contact number for all Macs are 4, and then for high quality Macs, the medium is 1 contact. So you can see we really obtained a very high quality results. And here I'm showing the number of Macs versus the total data generated for those 3 samples in comparison to the Zymo-Chill matrix standard, which is a human fecal reference sample. With the Zymo sample, so this one, we have reached saturation by sequencing on 4 Ravio cells to successfully characterize every single species of the strain. With the soil samples is a different story. And you can see that they're still very linear. So we're not accomplishing saturation. It means if you add more data, you probably will get more high quality Macs. So the Banfield sample is still ramping up, but it's probably getting close to saturation. We just don't know yet. And the reason for this is the different species diversity in each sample. So the species diversity of human gut reference is relatively low. It's only about 600 or so, but the Banfield wetland soil is medium, and the 2 DSMZ samples are in high and very high diversity. And so here we have provided the latest publications on long-read metagenomics. So the top one is a review paper covering both PacBio, Hi-Fi, and ONT sequencing. And the bottom one is our preprint discussing the current best practices for Hi-Fi metagenome assembly. And finally, we have all these digital available resources for your reference, and you can access all these resources at our website, pacb.com. So with that, I want to thank you for your time today, and we're happy to take any questions.
Thanks a lot, Ting. Very nice talk. Yeah. So I will start with my first question. In the 16S highlight, the highlight can use for the kinase or the standard protocol. They're no different. We have to adjust something.
Well,
I mean,
so
like
you have to.
if I sequencing from the library from kinase, or I sequencing from the standard protocol.
Oh,
link,
yeah.
right?
I got your question.
Okay.
No, you don't have to. You don't have to adjust anything. The Kinect sample, you just need to resegmentation step to break the long Kinect reads into short 16S, and then you can input those 16S like standard one into this pipeline.
Yeah.
You have to do one extra step.
Okay. Thank you. So and then my second question, so do you suggest us to using the meta NDBG rather than the hi-fi
ASM meta.
Yes.
So from our comparison study, the two assemblers are actually they're comparable. The results from them are comparable. Sometimes hi-fi ASM meta is better. Sometimes meta MDBG is better. So depending on the different sample types. So
Okay.
I think our latest suggestion is you can use both. Both max assembled from those assemblers, and then you can feed those max and the context into our hi-fi Mac pipeline for the post-processing.
Okay. So and then what is the input requirement for the hi-fi Mac highlight? Because like, do we need to sequencing with how depth it is? I mean, the depth of coverage for the hi-fi Mac. Do we need very depth or because as you show in your talk, you have very different kind of coverage.
The input is just the output from your assemblers, but if you want to recover more max, definitely you need higher coverage of your data.
Yes. So I just want to asking that how much is it? How
I don't know. Depend on the diversity of your species in your sample, right? So like in our study for the human fecal reference, the Zymo one, they just have 600 species in that sample, and we use just the 4 smart cells to reach the saturation. But for the soil samples, we use the 4, but and you can see that the number of max is still increasing.
Okay. So it means like acceptable to sequencing. We don't need to do the mark, but at least we can binding them. No, but at least we can build up the context for them. So the hi-fi mark highlight can stop, no, not stop, like just limit to the context generate the context only. We don't need to go to binding and to closing the genome.
Like the hi-fi Mac pipeline, the input of that pipeline is the max, maybe the C max, the high quality max, and also those linear context. And the hi-fi Mac pipeline will help you to further binning, like to assign those linear context to whichever species belong to which max. Yeah.
Because like I asking because like we sequencing not really that, and it means that we cannot go further to binding and then finish closing the genome. So the pipeline can help us to stop early, like the context only in the context step or not, or we have to do the full of the pipeline, the full of the whole pipeline. Did you get what I'm asking? Sorry that I'm confusing you. Okay, so move to the question from Sir, please. Can you please directly asking Sir?
So in a scenario with limited memory capacity, so which method or best practice type would you recommend for metagenomics profiling? SORMAS, Diamond Megan, or Minimap Megan?
Oh, I don't have a quick answer for your question. Yeah.
Yeah, because memory consume a lot. So and then we don't have
That's true.
the big HPC over here. We don't have the small station. So then I think that the sound math is the best because I try with the sound math and it look okay. Yeah, and the sound math is not require a lot of, especially we do with the camera around 21,
Yeah.
21 or 35 is okay. Yeah.
Yeah, I agree because the diamond and Megan is based on the protein alignment, so maybe it will consume more memories.
Yeah. Okay. So are there any more questions, please? It's a very good opportunity here. We have expert Ting and also Ray come from PacBio, and they know everything about their pipeline and so on. So please taking this opportunity.
Yeah, I mean, if you have further questions, you may also pass your question to DK stage, and they can reach out to us.
Okay,
Yeah.
perfect. Okay, thank you, Ray, thank you, Ting, thank you, Cham from the guys second PacBio. So now we move on to the next platform, the ONT. Thank you a lot for your time. Thank you.
Thank you.
Thank you.
So
hello.
Mavis, please. So I would like to introduce Mavis Tan. He bioinformatician from ONT, the Oxford Nanopore platform. So and then he also have I forget the name of the guy from BCA. Yeah. So he will support Mavis to answer some questions and so on. Yeah. So now everything is your Mavis, please share your screen.
Okay, thank you, Dr. Lui. Please let me share my before I share my screen, may I confirm that can you hear me okay?
Yes, I'm hearing well. Yeah.
Okay, thank you so much. Please allow me to share my screen.
Yeah, I
May
can
I
see your
my
screen.
screen?
Yeah.
Okay, thank you so much. So hello, everyone. Thank you, everyone, for taking the time to join us. So I think I will turn off my camera because in order to ensure that throughout the presentation, the internet connections can remain stable. Okay, so again, thank you everyone for taking the time to join us. I would like also to extend my gratitude to the organizing committee, Dr. Loy and his team, for inviting us to share the applications of the microbial genomics with Oxford Nanopore. So my name is Mavis. I'm the clinical sales specialist at Oxford Nanopore Technologies. And today I'm very exciting to discuss how Oxford Nanopore is shaping the future of microbial genetics. And also joining me is my colleagues, 10, a few application scientists from BCE Vietnam, who is our channel partners in the country. And throughout the presentation, please feel free, to type any questions that you have in the chat, either in English or in Vietnamese. And my colleagues, Tan will help to assist in answering the questions. So let's start with the overview of Nanopore sequencing for microbiology and infectious disease. So our first Nanopore sequencing platform is transforming the microbiology and infectious disease application. Our platform provides a comprehensive and versatile solutions for uncovering the microbial world with the application range from whole genome sequencing of the microbial isolates all the way to metagenomics. And let's break down into key application. First of all, microbial isolate whole genome sequencing, where this allows a rapid and accurate pathogen identifications during outbreaks and detect the resistant genes in real time. It gives us clinicians important information to inform treatment decisions. And additionally, the whole genome sequencing with Nanopore can also helps to understand the genetic diversity and also evolutions of the microbial populations, and it can guide the infectious control and the surveillance efforts. For full-length 16S, our technology enables the sequencing of the full-length 16S genes, and with long reads, it provides a high taxonomic resolutions. It offers a greater accuracy in microbial profiling compared to short read sequencing. And the ability to sequence multiple species in a single sample also allows for the detection of the polymicrobial infections, which are often missed by the traditional methods. Targeted viral whole genome sequencing and AMR. So with Nanopore, you can also classify both known and novel viral variants, which are essential for the monitoring of the viral evolutions, as what we've seen with the case of the SARS-CoV-2. And this can be applied in detecting the AMR as well. And by integrating the sequencing into the public health strategies, our platform actually can assist in tracking the viral outbreaks, guiding the vaccine strategy, and understanding viral transmission dynamics. And lastly, metagenomics. I think this is a very powerful tool to perform a microbial profiling because it doesn't require a prior knowledge of the pathogens. And at the same time, it also allows a rapid AMR profiling. So from the metagenome assembled genomes, MAX, you can assemble genomes directly from complex microbial communities, provide insight into the unculturable or the novel organisms. So what features of our platform make it powerful for implementations of microbiology and infectious disease applications? So first of all, our instruments are highly accessible. Historically, before Oxford Nanopore technology, owning a sequencer requires a significant investments. However, with Oxford Nanopore, you can start owning a sequencing capacity with just a few $1,000s. We offer the MinION, which enable a smaller labs to perform microbial genomics on site or even in field settings closer to the sample or outbreaks. And for the labs with higher throughput needs, we provide instruments like a GridION that you see in this image here, as well as a PrometheION series. And our library preparation protocols, such as rapid barcoding kit, are very streamlined and require very minimal hands-on time. If you combine this with the ability to acquire sequencing data in real time without the needs for the fixed runtime, as long as you gather sufficient data to answer your questions. So this helps in deliver a rapid turnaround time from sample to answer. And lastly, as you can see here, long-read sequencing technology often face some limitations compared to long-read sequencing technology, particularly when it comes to bacterial genome assembly. So short-read often results in a fragmented assembly with multiple contigs due to their inability to resolve repetitive regions, complex structural variants, as well as the genomic region with very high GC content. And additionally, bacterial genomes also often carry plasmid, which is the extra chromosomal DNA molecules that are crucial for antibiotics resistance and virulence. And also with short reads assemblies, frequently they will miss or misplace plasmid sequences, leading to incomplete or incorrect reconstructions. On the other hand, long read can capture the entire plasmid in a single read. This will enable a clear differentiation between the chromosomal and the plasmid sequences. So this improves the plasmid identification and characterizations, which is particularly important for understanding horizontal gene transfer and antibiotic resistant mechanisms. I believe many of you may know that Nanopore sequencing has been serving as a critical tool in pathogen surveillance. Even before COVID-19, Nanopore has already played a key role in global pathogens monitoring. So for example, two early high-profile efforts successfully used Nanopore for real-time sequencing of the Ebola virus in Africa, as well as the yellow fever virus in Brazil. And also to address COVID-19, the Africa CDC, in collaboration with Oxford Nanopore and other partners, have made significant investment to support the genomic surveillance across the continents. And this effort has led to the sequencing of over 100,000 of SARS-CoV-2 samples, with half of these 100,000 being sequenced with OMT platform. And additionally, more than 200 students and technicians were trained in the wet lab as well as the bioinformatics. And there are also many studies has now emphasized that the public health benefits of pathogen surveillance are greatly amplified when the sequencing can be conducted locally within the countries. So this has highlighted the importance of continued investment in pathogen surveillance infrastructures, such as the expanding of the local Nanopore sequencing capacity. And beyond the current pandemic, right, now that we are here, the building and maintaining a robust sequencing capacity for both known and emerging pathogens will be crucial in safeguarding the global public health. And by investing in local capability, we empower the nation to respond very quickly and decisively to the future public health threats. And before we jump into the technology side of things, I would like to share a few use cases that I think underscore ways the community is implementing Nanopore for rapid outbreak response today. So on the left-hand side, you can see that looking back to mid-May 2022, many of you may remember multiple cases of Mpox that started to be identified in several non-endemic countries. And on the May of 19th, Joanna and her team at the NIH Portugal have performed a rapid metagenomic sequencing with Nanopore. And this has resulted in the release of the first genome of the virus associated with this major outbreak, and then followed by the CDC using a similar approach on the Nanopore and upload the genome the next day. And in the late 2022, several European countries, including in the UK, have observed a surge in the case of the group A streptococcus. And the team at the Guy's and St. Thomas Hospital in the UK have been using the whole genome sequencing to study the bacteria isolated from the infected individual, where we will look at the case study later. Because by doing so, they aim to understand the epidemiology, such as the pattern, the causes, and the effects of these care conditions in the populations. However, I think the key point here is that they use on-site Nanopore sequencing, which allowed them to perform genome sequencing in real time as the outbreak was still ongoing. So this is in contrast to the traditional approach where sequencing might be done later after the outbreak has subsided. So the ability to conduct on-site real-time sequencing with Nanopore technology during the outbreak has allowed the team to act very quickly rather than waiting for sequencing results after the incidents. So this means that, they can potentially implement a faster intervention, improve the infectious control, and also possibly to reduce the number of cases or prevent the outbreak from getting worse. And finally, we also see that Dr. Eric Carlson in Cambodia, who reported the full viral genome sequence in less than 24 hours from a human infections with H5N1 avian influenza. So he actually leveraged this quick turnaround time with nanopore sequencing infrastructure that was initially put in during the COVID-19 time. And here we also see that there are many recent results from the community that have demonstrated like how our lattice chemistry is delivering a very high accuracy for a range of microbiology and infectious disease applications. So now that we have explored an overview of the role of nanopore sequencing in microbiology applications, now let's take a closer look at the technology itself. So what you can see here on the left-hand side of the slide, right? These are the surface of the floor cell. So it depends on the floor cell they are using. There are hundreds to thousands of these tiny holes, which is a nanopore, embedded in a what we call an electro-resistant membrane. And each of these nanopore will correspond to its own electrode connected to a channel and a sensor chip, which measure the electric current that flow through the nanopore. And during the sequencing, the electrical current will be applied. And when a molecule passes through a nanopore, the current will then be disrupted to produce a characteristic squiggle. From what you can see from this image here, this is what we call a squiggle. And then the squiggle will then be decoded using a base calling algorithms to determine the DNA or RNA sequence in real time. Or to make it easier to remember, you can think of the current as a water that flows through the pipes. So when an object enters the pipe, the flow of the water is disrupted, just as how the DNA or RNA disrupt the current as it's passed through the nanopore. So when a strand of the DNA or RNA that make up of a sequence of a different combinations of the nucleotide bases A, T, G, C. So when each base that pass through the nanopore cause the interruption to the signal, this characteristic interruption will then be converted to the base call. So this is this way of sequencing is what makes nanopore sequencing very unique, because it allows a direct real-time analysis of DNA or RNA, and including the modification informations, because when the base includes modification informations, the patterns will be different, and that's how our base calling algorithm are able to differentiate these signals. And what are the unique benefits of nanopore technology offers? So first of all, it allows the direct sequencing of DNA and RNA. So the benefits of doing the direct or native sequencing of DNA and RNA is that it can retain the epigenetic informations. And in fact, nanopore sequencing platform is the only sequencing platform in the market that allows you to do direct RNA sequencing without the needs to convert your RNA to cDNA with reverse transcriptions. So there are a few advantages of doing a direct RNA sequencing. So first of all, you get to study the epitranscriptomes, which is not possible with the cDNA approach. And also with the direct RNA, you can avoid the bias and errors introduced during the reverse transcription process, especially those with a complex secondary structures which may not be efficiently reverse transcribed. And in some cases, right, the reverse transcription failed to cover the full length of the RNA molecules, which result in incomplete transcript. So in addition to that, you also get the benefits for preservations of the strand information. And because of us being able to do a direct sequencing of DNA and RNA without the involvement of PCR. There's no amplification bias. It also means that the entire library preparation workflow is much more simpler and much more streamlined. And we are also read length agnostic, although we are often being perceived as a long read sequencing company. But the fact is that we also have the short fragment mode that is integrated into the operating software that allows the sequencing of fragment as short as 20 bases on the Nanopore platforms. So in other words, we can sequence as short as 20 bases to as long as you need for current records. The longest the single strand of DNA that is ever to sequence on the Nanopore platform is 4 MB. So it really depends on what is the the length of the library that you fit into the device, instead of having to limit what kind of length that you should put into the sequencer. And also we are the only sequencing company that allows you to do real-time analysis. So you can set like every half an hour or every 1 hour with the data generated, you want to do analysis and see whether or not the data generated is already sufficient to answer the questions. And if yes, you can choose to stop the sequencing run, wash the floor cell, and then keep it in the fridge so that it can be reused for your next experiments. So this is really a unique features that only offered by Nanopore technology. And also we give you the options for both devices and floor cell from a smaller format to a large format. So it means that you can run your experiment very cost effectively according to your sequencing needs. So in this slide, this is a summary of the relationship between our floor cell and device. Here on the left-hand side, the MinION and GridION devices runs 2 types of floor cell, Flonger and MinION floor cells. So the MinION floor cell have a theoretical maximum output of 48 gigabytes, and the scientific community have been always constantly trying to work towards this theoretical maximum. However, for now, you can typically the throughput that we observe is usually in 10 to 20 gigabytes, depending on a number of different factors like the sample quality, the quality, as well as the N50, etc. And then on the right-hand side, these are the Promethean series of device with theoretical maximum of over 270 gigabytes. And currently, you can expect between 90 to 150 gigabytes with the human whole genome sequencing. And we are constantly improving our chemistry, our floor cell to bring you to this theoretical maximum. And another thing to highlight is that it really depends on your throughput. You can start with a very small device running 1 floor cell at a time. And if you have higher throughput, you can have a GridION device that allows you to run 5 floor cells at a time. And for Promethean, we have Promethean 2, which allows you to run 2 floor cells at a time. Or if you require a higher throughput device, we have P24 or P48 that allows you to run 24 to 48 floor cell max at a time. However, one thing to highlight is that all of our sequencing device allow you to run each floor cell independently. It means that you can, for example, for P48, you can sequence anywhere from 1 floor cell to 48 floor cells without requiring any batchings. So in terms of the cost effectiveness or cost efficiency, you will not be getting penalized for not having enough samples to batch in a certain scenario. And with Nanopore sequencing, you can perform the real-time targeted sequencing through what we call adaptive samplings. or use this technique to reject targets that are not of interest. Like, for example, if you want to exclude the host genomic DNA in pathogen surveillance. So what is adaptive sampling? So this is actually a method which allows you to enrich or deplete a region of interest using software. So what you really need is a bed file which contain the region of interest and as well as a FASTA reference file. So once the sequencing started, right, due to the real-time nature of the Nanopore sequencing, our software, our operating software Mino, will be identifying and then to know that whether the strands that's being sequenced is within region of interest. And in 1 second for Nanopore platform, approximately 400 to 800 base pair of bases will be sequenced, depending on which device. And with real-time base calling and alignment, if the reads does not map to the region of interest you define, then the software will eject the DNA strains from the Nanopore. So then now the Nanopore will then be available for the next strand to come in. And the off-target strand are going to continually get rejected until your region of interest is being detected and the sequencing is allowed to proceed.
So just want to have a quick question. So this method, adaptive sampling can you for any of your machine or limited to some?
All of the machine. You can do it on MinION. You can also do it on the Promethean floor cell.
Okay, thank you.
Yeah. So with the adaptive samplings, you can potentially design an assay, one single assay that allows you to do SNP detection, allow you to do indels, allow you to do structural variants as well as methylation. Because with adaptive samplings, right, in terms of wet lab, you do not need to design any primers. There's no involvement of the PCR. There's no involvement of target capture with hybridization. All you really do is really just supply a back file and a FASTA file and telling the software which is your region of interest. So in terms of the workflow wise, it's exactly like whole genome sequencing, which is less than 60 minutes of the library preparation time, then you can do adaptive sampling. And also remember we mentioned about in the adaptive sampling, there's a lot of rejection that happens, right? And all these rejections that happens will be your off-target reads. However, the off-target reads can be useful as well because it can be then used for low-pass genome-wide applications. And one of the use case of these adaptive samplings in, for example, in human application is that you can design a targeted sequencing panel that have, for instance, pharmacogenomic genes or hereditary cancer genes. And so you will get a good coverage step depending on how many coverage step that you set, 30x, 50x. So you get that. At the same time, you also get this off-target reads for low-pass genome-wide analysis. Then you can actually do genome-wide imputation for PRS application. So this is really a very powerful tool that a lot of customers are very interested to optimize it to develop assay. And next, I would like to share with you some of the performance updates because, as you know, Nanopore is a very disruptive technology and is if you follow Nanopore development, you will see that it really develop and improve very fast and is still improving, every day. And in terms of the platform, sequencing platform, we actually cover the full spectrum of the entire sequencing needs in one single platform. So you don't need to buy, like, for instance, you want to do targeted sequencing, you buy a smaller device, or you want to do human genome sequencing, you need to get another big sequencer. With Nanopore, with a single platform, it's possible for you to do targeted sequencing all the way to telomere assembly. And now our new base caller latest updates is that with the super accurate base calling model, we can achieve Q26 simplex with simplex chemistry. And I mean, please bear in mind that when we say Q26 is actually referring to the raw read accuracy, which means the accuracy that achieved when reading a single DNA or RNA strand one time. Because most applications focus on variant calling or they report consensus accuracy or other metrics where the information from several reads is combined. So this can actually improve by increased raw reads accuracy, but can also be enhanced in another way, like for example, increased genomes coverage. And our indel, the F1 score improvement from 30x has approximately of 15% of reduction in error at 30x. And in terms of SNP, is still maintained at 99.9%, even at 20x coverage there. And in structural variants, we are also achieving more than 96% of F1 score, which is very good. And I think this one will be of interest of this group of audience. As we know, bacteria have a diverse modifications in a diverse context. And in the past, it can affect the base calling accuracy for native bacterial isolates and plasmids. So what we do is that our R&D have made effort to address this with a range of native and synthetic data training. And you can see in our previous versions of software, you can observe some sequencing error. But however, with the trainings on the native and synthetic data sets, you can see a massive improvement in the consensus accuracy for a diverse strands, including key human pathogens. And now we can observe more than Q50 of Nanopore-only bacterial assemblies. DNA modifications. So Nanopore is really like heading towards being a new, the gold standard of methylations or base modifications. We can do DNA modifications, including 5MC, 5HMC, 6MA, and 4MC right now. And all these can be done with low coverage there. And we have also constantly going to release more and more modifications. For example, now in development is this deoxyuracil, oxidation G, as well as the ribobases incorporation. For bacteria and viruses, the DNA modifications context is applicable as well, especially in the gene regulations, immune evasions, as well as the genome stabilities. So this is something you can look into. And in bacteria, RNA modifications can also play a critical role in gene regulation, in stress response, as well as in antibiotic resistance. And in viruses, they are key to replications and can be playing a role in the viral protein production as well. And therefore, the modifications are being increasingly studied for their potential in developing like the antiviral therapies and also enhancing the vaccine design. And as I mentioned earlier, I think Nanopore sequencing is still the only platform that are capable of detecting the direct RNA modification. For example, 6MA, pseudouridine, and we also have now the M6A plus inosine, as well as the M5C. And there are all also many more modifications that are going to be released with the ongoing and constant development and trainings of the new modification models. Additional capabilities will sure be released in future. So stay tuned for our development. So now let's look into the microbial isolate whole genome sequencing application on the Nanopore sequencing platform. So for this microbial whole genome sequencing, we have developed something called a NOMIS protocol. So it actually stands for Nanopore Only Microbial Isolate Sequencing Solutions. So with this approach, you can generate complete, high quality genome and effectively resolve the plasmids as well. And additionally, it also allows for accurate profiling of antimicrobial resistant genes. It offers you the informations into the resistant mechanism. And this method is very highly flexible. It can support sample batching. Or if let's say you don't require any batching, you can do that as well. And it also can deliver, rapid turnaround times, making it very ideal for both clinical and research setting, where speed and adaptability are essential. So I think we mentioned it earlier that one of the major benefits of Nanopore sequencing is the ability to generate a very long DNA reads, while the short read sequencing technology can give you hundreds of base pair long. Nanopore sequencing technology give you reads that are tens of thousands of base pair long. So this can come in handy when you are looking at the resistant genes, particularly if you want to look at or if you want to know something about where in the bacterial genome that the resistant gene is. And also when you sequence using short read sequencing technology, it can be hard to get this information because bacterial genome have a lot of repetitive elements. And short read sometimes is simply not long enough to cover these repetitive regions. So even if you sequence the whole genome, if you try to stitch the reads together and assemble them back into the original sequence, genome sequence, you also may end up with a bunch of short stretches of sequence, and you don't know how exactly where that fits. Where with long reads you can actually so with long read, you can actually, span this repetitive region, then you're actually able to recover a full circular sequence of the bacterial sequence as well as a plasmid. So instead of for example, knowing a bacteria carry a certain resistant gene, you can know where in the genome that gene is. So this is the overview of the end-to-end microbial isolate whole genome sequencing protocol. So this is a very flexible approach that allows you to sequence up to 24 microbial isolate genome per mean ion flow cells and generating a minimum coverage of 50x per genomes. And this 50x coverage threshold is what we tested and validated that is sufficient for downstream analysis, including accurate assembly, and accurate plasmid resolution, AMR profiling, as well as the SNP genotypings. And you can also get access to analyze your sequencing data using our software Epitome, which is a very user-friendly bioinformatics tool. So let's look at the sample preparations. So in terms of the sample preparations, we have developed 4 optimized extraction methods to generate high quality DNA from your cell cultures. So it really depends on whether you are looking into doing a universal applications on bacteria, fungi, or yeast. Then you can follow this, this extraction method to get a average sequencing read length of 4 to 7 kb. However, if your research require more than 7 kb, then it depending on the sample you are working on, whether it's a bacteria or is it very hard to lyse organism or is it fungi. We also have the optimized the protocol, and you can follow this protocol accordingly. to get the high quality genomic DNA for sequencing. And on the right hand side, you can see here that we have already listed here the extraction kits that we use to optimize the protocol, as well as the sample input requirement, the expected yield, as well as the expected DNA integrity number. So for sample extraction QC in general, we typically use two methods for DNA quantifications, the nano drop as well as qubit. So nano drop, as we know, is a spectrophotometer that measure the DNA concentration based on the absorptions at 260 nanometer. This is very widely used because it's very quick, it's very easy, and it also provides both the concentration and purity of your samples. And it also measure important ratio like 260-80 or 260-30, which can indicate the possible contamination with protein or salts. And for qubits, this is different concept. It is a fluorometer, so it uses a fluorescent to measure the DNA concentration, and is much more specific. So it quantifies the DNA by binding to a dye that only fluoresces when it binds to double-stranded DNA, so making it highly accurate for determine the actual amount of DNA that actually present in your sample. So in sequencing, with Nanopore sequencing including, it's very important to use nano drop reading together with the qubit measurement for more accurate quantifications. So this is because nanopore reading can sometimes overestimate DNA concentration, especially if there are contaminants like RNA, phenols, or other chemicals that left over or carry over from the extraction process. So if you observe a very significant difference between nanodrop and qubit readings, this could be a red flag for you to investigate whether your sample contain impurities that may affect the downstream process. So in general, the pure DNA should give you A260-80 ratio of about 1.8 and 260-30 of about 2 to 2.2. And if let's say you get a much lower ratio, it also can indicate phenol contamination. But it also can indicate carryover of guanine or carbohydrates. And for long-read sequencing technologies such as those, for example, like including Oxford Nanopore, getting a long DNA fragment is essential. The longer your DNA fragment with, especially in Nanopore, we have no limits on how long is your DNA fragments. So the better the read length and the more accurate the assembly is, and it enables the structural variance detections. And there are a variety of methods available to do fragment analysis. For example, the gel-based analysis, or you can use bioanalyzer or tap stations or femtopulse. However, if you are expecting fragments larger than 20 kb, then femtopulse would be ideal. So let's go into the library preparation part. So when you get your DNA ready, right? In this protocol, we recommend 200 nanogram of extracted genomic DNA. With this method, it has developed the process up to 24 samples with a genome size of up to 7 MB simultaneously. So if you are working on a genome size that is like smaller or larger than 7 MB, then you can adjust the multiplexing accordingly, depending on, of course, a number of factors like, for example, the sample quality, your genome size, or the coverage requirements. So on the right-hand side of the slide, right, you can see that this is the overview of the library preparation process. And the process begins with the segmentation step, where the transposase will simultaneously fragment and then add barcodes to the end of the DNA in one step. So this particular step only take 15 minutes. And then after that, the bar-coded libraries are then pulled, followed by impure bits cleanup, then attachment of the sequencing adapter to the DNA ends. And this entire process only take 30 minutes. So once the library is prepared, you can then prime your floor cell, load the library for the sequencing. So as you can see, the entire library preparation process is so quick, so easy, and so straightforward that gets you ready to start sequencing in less than 60 minutes. So analysis. So you can sequence up to 72 hours. However, it really also depending on, for example, the number of samples that you want to sequence at a time. Because if you sequence just a few samples, then you don't need to let it sequence for 72 hours. You can maybe just let it sequence for 8 hours or for 24 hours. As long as you generate enough data, then you can stop the sequencing, watch the floor cell, keep it for your next run. So it allows you this flexibility with Nanopore. And for sequencing software and data analysis, we have Mino, a very user-friendly operating software that drive nanopore sequencing devices, like for example, including data acquisition, real-time analysis and monitoring, local-based calling, data streaming. It also allows you to set different running parameters according to your needs as well as applications. And for data analysis, we have Epitome. Epitome have many analysis workflow that you can use. And it can be run either on the graphical user interface or command line. And these are free workflow, so you don't need to pay for the use of this workflow. Like for example, for bacteria whole genome sequencing, you can use WF bacteria genomes for you to do bacterial assembly, the annotations, the MLST, as well as allows you to predict the antimicrobial resistant profile as well.
So so it's free of charge and also include how many CPU and RAM are only available to download, or we can run it in cloud, or we have to pay for cloud.
So you can run it locally. You can also run it in cloud. Both are free.
Okay. So it means that the computation on using the CPU and RAM also free, and storage also free.
Correct.
Okay, perfect. Thank you.
Yeah. So the Epitome platform, as we mentioned, is really a user-friendly workflow because we know the importance of having the user-friendly workflow for scientists to streamline the analysis of the bacterial genomes. I'm sorry, I'm not sure why is it keep going to the next slide. So this tool it allows for multiple features. This workflow it allows for multiple features that simplify the process of characterizing the bacterial sample, making it accessible even if especially some wet lab scientists that don't have intensive bioinformatics experience. We also have integrates the multi-locus sequence typing MLST, a widely used method for strain identification and epidemiology tracking. So this workflow automatically identifies the bacterial species and provides a profile for any species. Just I'm here, right? Yeah. It allows any species with the publicly available MLST profile. And here, this one is the antimicrobial resistant profiling. So this workflow also allows the identifications of both acquired resistant genes as well as the chromosomal mutation that mediates the AMR. And also, if you are working with or if you are working on the food safety, and if you are looking for a Salmonella serotyping prediction tool, feel free to reach out to us. We have the end-to-end solution using the FDA approved 602, which allows for precise stereotyping. And it also provides the prediction for subspecies and, the all H1 and H2 antigens. And these predictions are important for identifying for specific serotypes of Salmonella, which is critical factor in tracking foodborne outbreaks and ensuring the public health safety. So this here is a case study demonstrate the power of whole genome sequencing for outbreak investigation, as well as the antimicrobial resistant prediction in a clinical microbiology settings. So the author is from the Providence Health Care Labs in Canada. They apply Nanopore long-read sequencing to investigate a cluster of multi-drug resistant Shigella shonae and aim to genetically characterize the MR genes and support potential interventions. So what they do is that they have sequenced 56 isolates using only the Nanopore platform, and they have found a strong evidence of a clonal strand responsible for the infections. And also the key highlights here is that from the study is that there was a very high concordance between a phenotypic antimicrobial susceptibility and the genotypic prediction generated by the Nanopore sequencing. So this included the accurate detection of the SNP that confer resistance to the quinolones, a very critical antimicrobial drug class. And also this analysis has showcased the base level accuracy of Nanopore sequencing, particularly in predicting the antimicrobial resistance directly from the genomic data. So I think this is really a valuable work that showcase, in an outbreak scenario, Nanopore sequencing can allow for a rapid intervention and better informs clinical decisions. And here in this slide, this is a very recent blog from Dr. Ryan Wick from Australia. So he has done like a benchmarking using our new base calling model for Nanopore sequencing. And you can see that based on his benchmarking results, you can see that there is this significant improvement in read accuracy and even greater improvement in consensus accuracy. And this includes, in this list, some of the, like for instance, the E. coli or Salmonella enterica that has about 50 to 53% of GC content. And you can see that from his comment here, he said that, this is the biggest improvement that I've seen in a while. Most of these ONT only bacterial genome are now more than Q60. So next, let's take a look at the full-length 16S microbial identification, which is one of the most common method for rapid and cost-effective microbial identifications. So the 16S genes has 9 hypervariable regions. I believe many of us know level SV1 through V9, which are positioned between the conserved sequences. So these variable regions is very useful because they can serve as the species-specific signatures. So it makes them very valuable for the bacterial identifications and classification in the microbial community profiling. And V3 and V4, these two regions are very commonly used for general bacterial profiling because they offer a good balance between the resolutions as well as the coverage. And for V1, V2, or the rest of the hypervariable regions, they tend to be more variable, but it may offer a better discrimination for certain bacterial groups. So if we compare traditional sequencing methods such as Sanger sequencing and short read sequencing typically they targets only a few hypervariable regions, for example, V3 and V4. So this may limit the amount of sequence information available and reduce the resolutions for the species level identifications. So in contrast with long reads, such as like Oxford Nanopore, we allows the full-length 16S sequencing. We cover the entire 9 hypervariable region. So this approach will provides a higher taxonomic resolutions, and it allows more accurate species level identification, particularly for closely related organisms. So in Nanopore, we do have 16S barcoding kit available, readily available. So you can, if you want to multiplex samples, like for example, to reduce the price of sample, if you only want to do 16S sequencing and you are interested in genus level bacterial identifications, and why we say genus level bacterial identification is that sometimes even with the full-length 16S sequencing, it can be insufficient for species level identification. Because a certain bacterial species share a very similar or nearly identical 16S gene sequences. So this can make it, make it very difficult to distinguish between these very closely related species. So in this case, the whole genome sequencing or sequencing of additional genetic region, like for example, the ITS regions, will be very helpful for a more precise species level identifications. But however, if you want to do 16S sequencing, we have these available. So here you can see the overview information of this 16S barcoding kit. So this kit helps to amplify the entire 16S region, 1,500 base pair approximately, followed by the library preparations where the DNA is amplified using a specific 16S primer. And then the kit has a preparation time of 25 minutes plus PCR. And it also requires 10 nanogram of genomic DNA per sample. It uses the kit 14 chemistry, which is our latest chemistry. It also includes 24 unique barcodes. It allows up to 24 samples to be pulled in one sequencing run. And in the post-sequencing, the analysis can also be performed using epitomy. And there is a WF 16S workflow that you can use. So you can see that this is a very, very simple end-to-end workflow with extremely short hands-on time. So the analysis workflow, as I mentioned, the name is WF16S. This is again freely accessible. Can be used to analyze 16S. However, it can also be used to analyze 18S or ITS region if those have been included in your sequencing. So all the epitomy workflow are available in both graphical user interface or command line. You can run it locally in your own device. You can also use cloud. And there will be also a similar software looks, no matter is locally or analyze it in cloud, so that there's no confusions and it's user friendly for you. And with Nanopore sequencing, one of the very powerful feature that we have is that real-time analysis is possible. What this means is that it allows the workflow to run continuously as the sequencing data is generated, which means that you can actually visualize the classification of the reads and the abundance of species in real time, dynamically updating reports. So you can know that, actually I already have sufficient data. I can stop the sequencing run. I can preserve it for the next run. And of course, if you want to analyze the data at the end of the sequencing, wait for the whole sequencing to complete, you can as well. And our analysis workflow provides very intuitive reports with interactive graphic as well. So, it's easier for you to visualize the data. So here's a case study showcasing the Nanopore 16S sequencing using our lattice chemistry. So this has enabled a highly accurate full-length 16S sequencing for environmental microbiota profiling. So in this study, the author has used the lattice chemistry and achieved 99% of raw read accuracy. And also it demonstrates that here, both Nanopore and an alternative long-read platform provide a very similar microbial profile, while the short read has a limited resolution for species-level identifications. And they also find, and emphasize that the Nanopore sequencing has enabled a species-level taxonomic identification from the environmental sample, highlighting its accuracy and its applicability of microbial community analysis. And also, I think it's important to keep in mind that Nanopore sequencing has continued to improve its sequencing accuracy, and also Nanopore sequencing platform offers you a very cost-effective solutions, not only in terms of the sequencing instruments, but also with a lower running cost compared to other platforms. And it's also very highly flexible. You don't need to batch your sample if you don't need to, and therefore you have a faster turnaround time. And this is very critical, especially in clinical settings, because you cannot predict how many samples is coming in for today, and you need that sample as quickly as possible. And because of our ability to generate real-time analysis data, it is also very powerful, because you can fully, utilize the flow cell just to the point that you generate sufficient data for your analysis. So next, let's us look into a pathogen agnostic metagenomics application. So Nanopore sequencing is, I would say that it revolutionized a rapid, unbiased pathogen surveillance, because traditionally, surveillance method focus on the retrospective investigations of known or suspected pathogens. However, the future lies in agnostic sequencing methods that allow for a very broad real-time pathogen monitoring, identifying what is present in a sample without prior assumptions. Like, for example, the targeted surveillance will ask, is this in the sample? Where agnostic surveillance will ask, what's in the sample? So the ability, because of the inexpensive and it can be high throughput sequencing technology for Nanopore, we sort of paved the way for next generations of pathogen surveillance. And through the monitorings of non-pathogens and detecting the emergent pathogen at the same time, the metagenomics surveillance enhance this with the capability to generate a metagenome assembled genomes, which offer a more detailed transmission analysis and better antibiotics resistant predictions. So this kind of pathogen surveillance can be very useful across hospital or across water purification plant, in farm, or other high-risk location where emerging diseases are likely to originate. And again, one of the key benefits of Nanopore technology in metagenomic context is that it has unrestricted read length. So it can enable a better metagenome assembled genomes and rapid access to results. This also allows a researcher to quickly gain insight into the microbial community composition and functional properties, enhancing the real-time decision making in critical scenario. So here you can see this is the bandage plots. Compare the short read and as well as the Nanopore long read assemblies. So in contrast to a fragmented assembly with many shorts, disconnected or tangled contigs, you can see that Nanopore data resulted in a very well-connected contigs, which indicates a more complete and higher quality assembly. So in this slide, you can see that Nanopore technology offer rapid turnaround time for metagenomics. So in making it, one of the options that is very suitable for analyzing complex environmental or clinical samples. And these samples often have low amount of pathogens together with, high level of, materials, DNA material from hosts or other common microorganisms, and typically requiring a specialized method for microbial enrichment. And here in this slide, you can see there are various metagenomic methods that have been developed by our customers for different scenarios. However, they all share one common trait, which is a very short turnaround time from sample to answer. Like for example, for respiratory metagenomics, our customer able to detect respiratory pathogens and resistant within 7 hours. CISPA and SMART9N, they are able to detect viruses like Ebola, SARS-CoV-2, or Mpox within 6 hours. And with adaptive sampling, it can deliver results in 5 hours and can efficiently detect bacterial pathogen as well as antimicrobial resistance. So I think out of the different methods, let's review one of the metagenomic protocol developed by Adela, Professor Jonathan Exworth and colleague from Guy's and St. Thomas Hospital with the support from Oxford Nanopore. So this is the metagenomic methods developed for both DNA and RNA sequencing of pathogens. And this method can be applied to routine samples such as the nasopharyngeal swab or the bronchoalveolar lavage. And it has also successfully sequenced pathogens like the Mpox virus from the skin lesion swabs. So it demonstrates its versatility across different sample types. So you can try it. And this protocol cover the recommendation in the RNA and DNA extraction, followed by the cDNA synthesis using random hexamer and oligo-DT primers for the RNA in the sample. And then we just follow the the standard rapid PCR barcoding protocol, which we will need to tagment your DNA, and then you PCR both your DNA and cDNA strand. And then you just attach the sequencing adapters, supply in the kit, and then prime the flow cell, load your DNA library into the flow cell. And in terms of analysis, we have WF metagenomics for identifications of the original single reads from both amplicons, targeted or shotgun metagenomic sequencing. So it also provides you with the intuitive reports with interactive graphics. Again, you can run it in real time to see the composition of the microbial profiles in your sample, or you can do it post run with a final workflow that require more computing. So I think this case is a case study, the respiratory metagenomic workflow that we discussed earlier. So this Nanopore-based respiratory metagenomics, in fact, has demonstrates a significant impact in the medical field, particularly in the ICU settings. So this is a case study that highlights the potential of respiratory metagenomic in ICU, where the workflow that we see just now is a single test that can test for bacteria, for fungi, for DNA and RNA viruses. So if I were to put it into the perspective, you can see that here, the comment from the CEO of the Guy's and St. Thomas Hospital, that the ICU costs range from £2,500 to £10,000 per day, depending on the severity of the patients. Additionally, the use of the broad spectrum antibiotics has also continued to drive the antimicrobial resistance. Is a growing global threat identified by the WHO. And therefore, there is a very urgent need for a sensitive, rapid, and also less biased diagnostic tool for respiratory infections. And respiratory metagenomics so far has shown promise as a first-line diagnostic tool for lower respiratory tract infections, with the ability to detect, all the pathogens and offer resistant profile to assist with the infections control and also the public health measure. And however, the questions it still remains in what is the feasibility of doing same-day testing and whether the results are frequent and informative enough for routine clinical implementation. And that's the that's the reason why they have this pilot study. In this pilot study, they do this respiratory metagenomic workflow, and you can see here's the results. They have so far performed on more than 500 samples with 250 samples as part of the pilots. And the results has shows that 90% of the samples, they are able to get same-day results with the median turnaround time of 6.7 hours versus the traditional methods, culture-based method that takes 40 hours. And the sensitivity is 93%, specificity of 95%. And more importantly, you can see 45% of the cases resulted in informed antimicrobial prescription changes, and 20% escalation on the same day, 25% of the cases de-escalations mostly next day. And also, this study has also uncovered a hidden infectious burden in the ICU setting that are, often missed by, traditional testing. Like for example, one of the example is a cluster that involved 3 patients with 99.9999% of the genetic similarity of the Klebsiella varicola that was identified. So this incident has suggested there is a potential patient-to-patient transmission due to overlapping work stays. So these insights has offered the potential for improved infections control, and it also can prevent further escalations if they, handle this issue more in a faster manner. So given the recognized, gap in the preparedness highlighted by the COVID-19 pandemic, as well as the increasing antimicrobial resistance, which is a global issues, this study actually gives both encouragement and the urgency to introduce metagenomics for evaluation as a standard of care in acute care pathway. Especially in the ICU setting, every hour counts. If there's any improvement made to the treatment management, it can save life. So this is my last slide. Thank you for listening.
Thank you so much. Very nice information. Thanks a lot for a very nice talk. Thank you. So I will start asking first, and later we will have more questions to come. So can you come back into the last slide?
Okay, sure. Last slide. Oh, I need to share my screen.
Yes, please.
Last slide is It didn't go back to the last one. This slide?
Yes, this one. Yeah. So so the pipeline for the laboratory on the left hand side, at the bottom of the left hand side, we can see that for the microbiome detection, it's around 6.5 hours. And then after we identify which microbiome, and then we detect the AMR 2 more hours. So in total, we have around 8.5 hours, isn't it?
Yes, I think this is a timeline that in average from the you get this sampling, and the median turnaround time is within 6.7 hours. You're able to detect the microbial. With sometimes you already have the enough data for MR detection, but sometimes you may need a little longer time for a more precise MR detection. And by 16 hours, you get the whole genome being assembled. So you get all this information before your standard, 40 hours cultures data come back.
Yeah. So and then my question is, this one, do we apply the adaptive sampling into it? Like we give a set of the AMR gene database for the software in order to do something like targeting them or
No, this is a metagenomics, so they don't do any adaptive sampling. So they will find if anything that's inside there, regardless of whether it's bacteria or fungi or DNA or RNA viruses.
Okay. So I just think that they will do some kind of adaptive sampling that give a list of on the AMR gene into the software, and then if they detect them, and then they do with sequencing deeper for the gene, otherwise they don't. So it's not. So we sequencing everything.
Yes, in this case, they're sequencing everything.
Okay, perfect. Yeah. So then we have some questions in the chat box. The first one is how does the real-time error correction work, especially give the high error rates associated with the Nanopore data?
Let me see the chat.
Yes, it's very beginning of your slide. So you mentioned something about the error correction. Yeah.
Okay. All right. To answer that questions, right? Indeed, if let's say it's a real-time analysis, right? Because we have different workflow that you can select, or different tool that you can select. With the real-time analysis, of course, the accuracy you may have a higher rates because the accuracy is lower. But it allows you to sort of like knowing whether you already have sufficient data to stop the sequencing run already. And you can actually then do another analysis with the better or finer workflow that give you a more accurate or higher accuracy data sets. And I think when it comes to because if you are familiar with nanopore sequencing, you'll know that there are 3 types of base calling model. Fast, this one will use the least computing power. It will enable you to get the sequence fastest. However, it also has a higher error rates. And then the second mode is high accuracy model. The third one is super accuracy model. But however, because the bacteria genomes are typically very small, so there's between fast, hack, and sub, there's not much differences in terms of very, very significant in terms of the computing capacity required for between hack and sub best calling model. Mm.
Okay. So and then the next question is about what sample reparation consider are essential for detecting the AMR gene effectively with the nanopore sequencing, especially in the complex microbiome.
I think it is for microbiomes, I think it depends on the sample types, like whether it is from soil, is it from stool, or from different types of microbiome. I think one of the challenge remain on extracting high quality um, genetic materials from all these samples. So you may need to try different extraction protocol. So in Nanopore workflow, in Oxford Nanopore, right, we actually, in our website, there's one section that has extraction protocol. You can actually search, depending on the sample types that you are working on, you can try to search whether or not there's a collection or the past work that have been done, and then it's showing good result with the Nanopore platform. Then you can actually try that particular extraction methods to ensure that you get good the good material to start with. And
How about the
providing sorry
the blood one, the raw paper blood.
blood. Yeah,
Yes.
blood is very common. It's very, very common. We do have that protocol in our website.
Okay, thank you. And the last question from Huy, he asking, can ONT real-time analysis capability to be applied to the AMR detection for rapid point of care testing? If so, what are the main limitations?
I think definitely yes. I think, but of course it depends on your definition of rapid point of care testing. Are you saying 45 minutes or 30 minutes or 1 hour?
Yeah.
But no matter how we have use cases of adaptive samplings that are able to diagnose a newborn within 3 hours of birth. So I would say that it is possible, like for example, using adaptive sampling, as you mentioned, Dr. Loy, that you have this idea of detecting the MR with adaptive samplings. I think that can be developed definitely. And the main limitation is that with adaptive samplings, you do need to ensure that the computer that you have has the enough or the sufficient computing power to catch up with the adaptive sampling, because it does require a little bit more of computing required than the normal sequencing.
Yeah. And I just want to asking about the CIVD because we want to apply in a hospital about healthcare, and then we have any available solution for the CIVD.
Okay. Our future path, for Nanopore is definitely one of the focus is on the clinical application. So a lot of customers using us for clinical LDT. However, for IVD, we do have our devices, GridION, that are going to have Q lines. And after the Q line approval, we are also going to submit for IVDR. So in terms of the IVD kit, we are partnering with Biomaru. So we will have one ample TV kits that are going to be launched with Biomaru very soon, maybe end of this year or early next year. But please stay tuned with the release of our first IVD kits. And with that, there are also some applications that we are developing to add more and more menu under the, for diagnostic use after the TB. Yes.
Yeah. Thank you a lot. Are there any more questions, please? It's very nice talk from Tan. Very nice.
I have one question for the wet lab protocol.
Yeah, sure.
In the QC step, if I use some kind of kit for size selection, then should I use uh, uh, staff station or bioanalyzer to QC the symbol again?
If I understand you correctly, your question is asking whether to use bioanalyzer or tab station for QC, right?
Yes.
Oh, it's fine. Depending on your labs, whether bioanalyzer, you have bioanalyzer or you have tab station, both are fine.
I mean that if I use the protocols, if I use some kind of kit like some kind of kit that used for size selection of the fragment DNA, then should I use staff station for QC my symbol again?
Yeah, sure. I think it's optional, but you definitely can, because it will give you the you will visualize, how effective is your size selection. You can see whether or not there's, any smaller fragments or is the profile what you are expecting. It's optional, but it's good to have.
There are any requirement for DNA for distributor of DNA fragment when I input the symbol? I mean that I gonna sequence whole genome sequencing, then should I then do there are there any requirement for distribution of DNA fragment?
Do you specifically refer to this one? For this one, the NOMIS protocol? Sorry. You're really referring to this one?
Yeah.
Oh,
Suppose that
it's not. Yeah. Okay. Yeah. Okay. It's not required. So it's not compulsory for you to to really need to do the fragment analysis before you start sequencing. It's optional.
Yeah, it's optional, so you don't have to.
Yeah,
Thank you.
you don't have to.
So
Yeah.
then we have Paul. Are you okay with the answer, or you want to asking further?
It's okay.
Okay, thank you. So we have one question from you. Are multiple enzyme rapidly sequencing adapters? So I even don't understand the question. So can you please elaborate your question, please?
So in the I see in the slides, the purple color mean the rabbit sequencing adapters. Is that motor enzyme?
Oh, okay. So that motor enzyme, right, is important because it will help to control, the movement of the DNA or RNA when they go through the pore.
So
You're asking that the motor enzyme in the sequencing adapter?
yeah.
Yes, they are together. The
So
rapid sequencing adapter.
yes, is that the same enzyme?
Same enzyme.
So on the right side, the purple color, I mean, is that the motor enzyme you
Excuse me, maybe I can help to address this question. So allow me to explain it in Vietnamese for Miss Dương. Chị Dương ơi, câu hỏi của chị là cái phần motor enzyme và phần sequencing adapter ở trong biểu đồ đúng không ạ? Thì
Đúng rồi.
ở trong biểu đồ thì mỗi cái sợi DNA hoặc là RNA sẽ được gắn một cái phần adapter, và motor enzyme là cái cục màu tím thì nó sẽ bám vào cái phần adapter đấy. Ờ,
Đi. Cái motor enzyme đó là nó nằm trong cái kit chuẩn bị thư viện của mình đúng không?
đúng
Hay
ạ.
là nó nằm trong cái flow cell
Nó là một thành phần trong kit chuẩn bị thư viện ạ.
là nó gắn vào adapter?
Vâng ạ.
Ok, cảm ơn em.
Vâng ạ, vâng.
Okay, thank you. Thank you, you. So, please, if you have further question, you can send an email to BCE over here. We have Thanh Dương, who is the representative for BCE in Vietnam. Yeah. So, mọi người mà có thêm câu hỏi gì thì ở đây có bạn Dương, bạn Thanh Dương á, là đã gỡ cái contact ha. Thì BCE thì là cái nhà phân phối Oxford Nanopore của Việt Nam mình ha. Thì mọi anh chị em đây có câu hỏi thì cứ thoải mái ha, gửi email đó contact cho bạn Thanh Dương ở đây nè, có địa chỉ email rồi mọi người. Rồi, ở đây còn có ai có câu hỏi thêm nữa mọi người? Are there any more questions, please? Các bạn có thể hỏi tiếng Việt rồi xong xuôi đây có Dương nè, rồi có tụi tôi nữa, tụi tôi sẽ giúp dịch sang tiếng Anh. Mọi người thoải mái là mọi người. Ở đây là cơ hội của mình, không tốn tiền, không gì hết, không hỏi. Chi vậy, đúng không? Đó, mình nghe, mình đã dành tới thời gian này rồi thì mình phải tận dụng vào. Đó, người ta nói một cái bài quá trời hay luôn, quá trời thông tin luôn. Không biết cái gì thì hỏi đi, chứ mốt xong rồi tan rồi thì hỏi đâu có ai nhớ, đúng không? Đó, hỏi đi mọi người, không hỏi thì tan á. Tôi cũng buồn ngủ lắm mà, nhưng mà tôi cũng muốn đã làm thì làm cho nó tới tới nơi tới chốn chứ. Có ai hỏi nữa không? Rồi không ai hỏi nữa thì thôi nha, tôi kết thúc đó, đừng có hối hận nha. So
Cho Nhân hỏi một xíu là cái slide cuối mà bác Maviston trình bày á, nó khá là hay, nhưng mà không biết là áp dụng ở các bệnh viện ở Việt Nam mình á, thì chi phí cho một bệnh nhân như vậy á thì nó có khả thi không? Cái này không biết hỏi ai.
để mình hỏi. Mavis, please
Yes.
re-share your slide. So she asking that the idea of the method using the Oxford Nanopore method to reduce the time is very good. So she just want to asking that it's possible to apply in the clinical setup in Vietnam. She want to know the code. How
Which one? Which slide?
the last slide.
Oh, the last slide.
Yes. That's why I
Okay.
asking, yeah, about the using the orthogonal to discover the detection the bacteria as well as the AMR for lungs infection disease.
Yes, so for implementation of the, in fact, this particular study is in ICU settings, is clinical.
Where do you know where it happened?
It is in the Guy's and St. Thomas in the UK.
Okay.
Yeah, so one of the PI, his name is Professor Jonathan Exworth. So he's one of the lead PI for this program. He is also currently working with Nanopore part-time as a medical affairs. You know, if you are interested to bring into your, you know, your hospital, very happy to connect you with Professor Jonathan so that you can, you know, discuss this in further details on, you know, how can you actually bring this as well. And in fact, He's here in Asia a few months ago, and he's going to be here in Asia as well. But maybe this trip, he will not be covering Vietnam, but the subsequent trip, he can actually visit Vietnam and to discuss this in further detail with you as well.
Thank you. So because we don't see any publication, so we rather to read the publication first and then.
Oh, there is a few. I share with you,
Okay.
and you can share with the rest of the group. And
Okay.
like for example, this one just got published in 2024, and please feel free to read it, because this is a very detailed articles. And if you go to the supplementary, the data, they also describe in details on how they actually do this. Is very, very good reading. Yeah, please. I'll share this with you.
Okay.
It will
Thank you.
describe in details what have been done.
Yeah. So I refer to analyze the data to see how it look like first, and then we were thinking about asking further information. Yeah.
Yeah, sure.
Because
Feel
the
free to reach out to us, or BCE, and then they will reach out to us, and we can always meet.
Yeah.
Can I please have one question?
Please go ahead.
I have the mean ion, and then it says that it's just the mean ion is one flow cell, and then the maximum of the sample we can run on the mean ion for one flow cell. Is it depending on the kit I use, like 24, the kit with 24 barcode or kit with 96 barcode? So what's the maximum sample I can run in at one time?
Can you repeat your question again? What I get is that how
So,
many samples can be run in
yes.
one flow cell, right? For which
Yes.
application?
For whole genome sequencing, like to close genome.
Oh, okay. So for whole genome sequencing of bacterial isolates, the NOMIS protocol that we have optimized end to end, that one allows up to 24 samples to be multiplexed in one flow cell.
In one flow cell.
However, if you have less than
in
that, let's say you only have a few samples, right? You
Yeah.
don't need to run it for a full 72 hours. You can actually run it shorter, and then once you generate, you get enough data, you can actually stop the sequencing run and then use the flow cell for next run. But if you have a full 24 sample, you can let it run for a full 72 hours, and then at the end of the experiments, you will get all the data that you need. So with Nanopore, you always have this flexibility. So it will be very subjective to answer, how many samples to be multiplexed in a run, because it depends on the number of samples that you have, the coverage depth that you require, and the application as well.
Yeah. Thank you.
In terms of barcodes, you can multiplex 24 or 96, and even there's a publication that published last year in 2023. It is a article that is describing how they actually barcodes 100K specimens in one single nanopore runs. So you can do like a dual index kind of things to increase that barcoding multiplexing capability. Yeah, so a lot of things can be done on this platform.
Yeah. Thank you.
Yeah, you're welcome.
Thank you. So any further question, please? Okay. If there is no more questions, please. We have the link for register for next lecture. Thank you. Thank you again for Mavis and as well as Ray and Ting. And also thank you for BCE and BKSH that helped us to invite very good speaker. If you have any further question, please send the email to the BCE or the BKSH. They will transfer the information to the speaker today. So
Yes.
thank you again for a very nice talk, very nice information. Yeah.
Thank you, Dr. Lui. Thank you so much for everyone who joined. Thank you.
Thank
Thank you,
you.
thầy Lợi. Thank you, everyone.
See you next time. Yeah.
Thank
See
you.
you.
See
Thank
you.
Bye bye.
you.
Um,
Thank you.